And one more note, if you want to leverage the closed captioning and have transcript available there is a closed captioning button you can activate during the talks if you want that on in the background. And then we'll do a quick intro one last time for anyone just joining before we get started. We are waiting to get started at 9 a.m. You can see the agenda on the screen. We will be starting with talks from the team from the Allen Institute introducing the OpenScope opportunity. This will be an overview of the program and the mission of the OpenScope program, the RFP that has been posted, and the application process, as well as the future of OpenScope. Then we will go on to some team members outside of Allen Institute by both the DANDI archive and Neurodata Without Borders. We are not taking questions during the talks, but there will be time for Q&A after the end of each talk. And if you have questions, you can enter them in the Q&A function within Zoom, so we can get to those during the Q&A portion. If you enter in the chat, they might be overlooked, so just make sure you're entering them in the Q&A, not the chat box. You can also, we will be taking any questions and comments from the YouTube and forwarding them here as well. And just one final reminder that this is being live streamed on YouTube and will be recorded and posted in YouTube for posterity. So it is being recorded, and you can always refer back to it after the live webinar today. If you requested an individual consultation during your registration for the webinar, you should have received a separate email with a link to where you can book a 10-minute slot with the Allen Institute staff. And if you haven't done that yet, we're still taking slots for this week, and reach out to us if you did not receive that and you requested a one-on-one consultation before September 7th. Any other questions, you can always email us at openscope@alleninstitute.org. And you can always check the website for the OpenScope with the RFP is posted with details on the opportunity there. We will be beginning in just a few minutes. Okay, we're going to go ahead and get started. So welcome everyone to the first OpenScope scientific webinar. This is the first year of the new OpenScope program, and it's a goal of opening up some Allen Institute resources for the Allen Brain Observatory, from a survey-based model that we use the Allen to opening up to more hypothesis-driven experiments from the community. My name is Carly Kislisnik. I am a project manager and alliance manager at the Allen Institute within the MindScope program. I will be helping manage this OpenScope program and also moderating for today. Our agenda for today, we are going to first have two of us from the Allen Institute, myself and Jerome Lecoq, introducing the OpenScope program, the mission and why we are doing this, as well as some past projects we've done. We're also going to talk about the RFP, or request for proposals, that was just released, where you can submit proposals in your planned experiments and the application process. And then also talk about the future and what we see for the next few years in OpenScope. We'll have time for some Q&A, and then we're going to open it up to two talks from other people in the community outside the Allen Institute. We'll talk about the DANDI archive for an online repository for neurophysiology data by Satra Ghosh. And then move on to a talk by Neurodata Without Borders on the data standards and the NWB files by Oliver Rubel. We will have time for Q&A at the end of each of these, and then also more time at the end for general Q&A on any of these topics. If you have questions throughout, use the Q&A box on Zoom and add your written questions. We won't be taking questions during the talks, but afterwards we'll look for the written questions. And just make sure you're not using the chat function as opposed to the Q&A box. If you requested a individual consultation when you registered for the webinar, you should have received a separate email from us to schedule a 10-minute slot this week. Please reach out to us if you did not receive that. We are still scheduling them for this week. And any other questions, we encourage you to reach out to us to discuss your planned experiment at openscope@alleninstitute.org, and also refer to the website where the RFP is posted. Just a reminder, this will be live streamed on YouTube and recorded and posted there for posterity. So you can always refer back to this if you want to see the webinar again. So we encourage you to use that resource. Okay. So really quick, we're going to start the OpenScope session from two of us from the Allen Institute. And so we're going to talk about first the OpenScope program and our mission and plans for this new model that we're trying to model an observatory in systems neuroscience. We're going to talk about how this builds off of the existing Allen Brain Observatory pipeline. And then we'll go into the specifics of the 2021 request for proposals for this opportunity and the application process. Then we'll just discuss the future of what OpenScope can look like and the developments and outlooks in future years as well. I'm going to go ahead and hand this off to Jerome, who is the principal investigator for the OpenScope project. So Jerome, if you want to take it from here.
Hello, everyone. So my name is Jerome. Together with Christophe Koch, I've been leading this project in the last few years. And so we are extremely excited to have this webinar with you. We'll start this talk with a little bit of background on what OpenScope is and why we are doing this in the next few slides. So if I could. So why is this interesting to sort of dive into this model? So the inspiration for OpenScope really came from astronomy. And this was something that Christophe Koch brought with him at the Allen Institute and was sort of a long-standing view of what we could bring the field forward. And in these slides, we have an illustration from the Keck Observatory in Hawaii. So it's a pair of telescopes watching the night sky. And you see that this model of sort of shared facility with scientists across the world has been extremely successful. This is the number of publications of the years that are sort of using in some ways the resources from the observatory in astronomy. And you see that sort of is kicking off really quickly and having a tremendous impact on its field. And so we thought that there is a possibility here that this model could be equally impactful on our field, and we thought we should try it. So obviously, there is some similarity with astronomy. There's also some differences. And on this slide, we highlighted some sort of shared principle as to why this could work. The first rationale is that we know that the brain is complex, and it's a very hard piece of matter that is difficult to understand. And so to tackle this, we need a large number of experiments, and this experiment needs to be integrated. And so the status quo of the field is that a lot of labs are independently pursuing to understand the brain, but it's really important to enable some integration of this data set. And so we hope that an observatory model could help alleviate this issue. The second challenge is that some of these experiments are sometimes duplicated. It's important to duplicate research to validate results, but sometimes it's a waste of resources, or it sort of prevents us from doing bigger and more impactful science. And a last point that might be actually very important, it's dear to me, is that there are multiple communities in neuroscience. And there's an experimental community, a technological community that is developing the tools, and theoretical neuroscientists that are asking hypothesis-driven questions on how the brain might work. And we think that an observatory model could help integrate and synergize this community together by having this central place where ideas can be discussed and compare one another with the data that is acquired. So what were sort of the background ideas that brought us to test this model? So OpenScope is, so we started OpenScope a couple of years ago using Allen Institute Fund as a pilot to see if there was interest in the field. But it is now starting from this year and NIH U24 resources. So we are extremely thankful for the NIH vision and support to let us try and support this model. And so this is going to be a resource for the next 5 years with a yearly review. So we have to be fulfilling our milestones. And it's also part of the Allen Institute Mindscope program. So at the Allen Institute, the Mindscope program has a portfolio of research projects, and the OpenScope project is one of them. Our goal is to open the Allen Brain Observatory 2-photon and NeuroPixel experimental platform for experiment proposed by the community. So that's is exactly an observatory model as in astronomy. Obviously, there are other long-term goals. One of them is to maximize the impact of our platform to help them succeed and have more sort of scientific knowledge generated by them. But it's also to build a community around it, and this will enable to sort of develop the system neuroscience ecosystem, which is why today, for instance, we have representative from the NWB and the DANDI archive project, because they're part of our ecosystem and we want to help develop it. This is a good sort of proxy into one thing that is really important to me is a couple of guiding principles to this project. And we want to be accountable to them. The first thing is we are an observatory for the community. So we are meant to serve you all. And so our goal is to empower the neuroscience ecosystem, not replace or rebuild it. So there are existing tools, and whenever possible, we will adopt them. As I said, we have a representative from 2 projects, but there'll be more in the future, I hope. And so our goal is to synergize with the community whenever possible. And a last guiding principle that is really important, and you know the elements should follow them, is open source, open data, and standardized protocol is really core DNA. And so one of our goals with this resource is to help encourage and support its practices throughout the entire field. So based on these resources, this is sort of the component of the NIH U24 resources. So the first thing is access to the LN Brain Observatory end-to-end pipeline. So this is coming from surgery to data generated in the cloud. And so we are making this available for experimental proposals. This proposal will be selected through a yearly cycle with double-blinded review. So that means that we will make our best to have no bias in the selection except the science. It's a science first and foremost. And one of our important components, this is open to the entire community. So we are really talking about the entire world here. And this is sort of the same way as astronomical observatory are sort of open, and it's made free. A third component is an integrated cloud data deployment analysis ecosystem. And so this will be made in collaboration with first the NWB, Neurodata Without Border format, which is a way to standardize neurophysiological data storage. And the DANDI archive, which is the place where this data is stored and will enable an ecosystem of analysis routine in the future. And the last component, but not the least important, is a technical hub. So any observatory is there to serve its community and also to disseminate the tools and technology that we develop. And so we'll have 2 opportunities for that. The first one is one that we have today. And this is it's a little bit of a special webinar because it's the first year and we are starting the project, but we hope in the year to come, we'll have more scientific content in this webinar. So this is an annual scientific workshop. And the second is an on-site technical workshop. Well, some people, maybe some of you could come on-site and see how we are actually acquiring the data firsthand. And so this will something you should keep an eye on because it will be coming in the next few years. There will be a number of proposal cycles in the year to come. So we have 4 cycles of our plan, and we'll come back to the slides multiple times in the talk. But the idea is that at the end of the NIH resource 5 years, we'll have executed 14 projects. So now that you sort of know a little bit the main component of the OpenScope resource, I'm going to spend a little bit of time talking about what is the LN Brain Observatory pipeline, because there's a lot of background there. I'll discuss about the technical components and what we've used it so far. So the LN Brain Observatory pipeline is, as we said, an end-to-end data collection pipeline. You start with transgenic mice and finish with data products that are released to the community. There are several key components to it. And our goal with the OpenScope platform is that you somehow don't have to worry about the technical details, but now I'm going to talk about them so that you know what you can rely on. So there are transgenic mice, as I said, so these are bred in-house. Some of them have calcium reporters, some of them have opsin expressed in them. And they are sort of going through a surgery. So we have a dedicated team that is doing surgical implant, be it glass window or head fixation apparatus, so that we can record with high stability and confident about the units. So that team performed the surgery, and then we go to a separate team that is mapping the visual cortex using intrinsic imaging. So that takes a week of work. And so that gives you a sort of a coordinate access about where the different brain area are on the dorsal part of the cortex, the visual areas. So we map a certain number of them. And then in the next step of the pipeline, animals undergo either a passive habituation to head fixation, and then habituation to passively viewing stimuli, or behavior training. And right now we are doing change of detection training, but we hope in the future that's going to sort of expand. So once the animal is either habituated to passive viewing, so this is done on a separate cluster. They're transferred to our sort of neurophysiology pipeline. And so there are three types of it. One is a single plane imaging, so to record with calcium imaging the activity of individual neurons with a single plane at a time. Another one is multi-plane two-photon calcium imaging. So it's a technology that we've developed in-house, and I have the slides to show it to you. And the last one is neuropixel recording. So this step allows to record the activity of the neurons. The data is acquired, stored internally, and then will be converted into NWB files. Then the animal is perfused, and we extract the brain to check for brain health, and in some cases, extract the coordinate of the neuropixel probe track. All the data are going to data processing, and then in the last few years, we have made available to the community a web product, which we'll talk a little bit about. All of this is done with high level of standardization. So here I'm just highlighting one aspect, because there are several to this. The first aspect of this is that we have a sort of a intimate knowledge about the rig geometry. So here you see a CAD rendering of the NeuroPixels platform, and the two-photon platform is just nearby. And so you see that they share a very high level of standardization. And throughout the whole pipeline, so this starts from the surgical implantation to how we monitor surgical location, as well as how we do intrinsic imaging and neurophysiology. We have a coordinate system that allows us to keep track of where we are recording from. And this allows us, using intrinsic image here, to map precisely the recording location. I encourage you to look at this paper put together by Peter and David recently, to get more details about this process if you're interested. And we also have shared the hardware that is underneath this system. So a tremendous level of standardization, but that is made available to you all. Once that sort of imaging location is determined, we can use, for instance, multi-area two-photon calcium imaging. And so this is an instrument that allows to record up to 8 planes throughout the visual cortex, and we hope to do sort of a broader recording later on. And here's an example where we're recording from 4 different layers in V1 and LM at 10 Hertz, the volume rate. And there's more information as well in this publication that was put together by Natalia, Dimitri, and Farzaneh and Najafi last year. And we actually made an update this summer to this paper. As well as sort of multi-plane calcium imaging, we also have the ability to do neuropixel recording. And so this is a system that we can position 6 probes throughout the visual cortex. Currently, the location of the probe is fixed to be very precisely over the visual cortex and some subcortical visual areas. There's also like a publication that sort of highlights this platform. That was put together by Josh, Severin, and Sean Olsen, as well as a lot of other people at the Allen Institute. And so you have the yield here that is sort of highlighting where we can record from. And so you can use this to see individual spike during the visual stimulus that you will design. Yes. And so I'm not going to spend too much time on the behavior aspect because this year is focused on passively viewing. But I do want to mention that we spend a lot of effort in sort of developing a detection of change behavior at the Allen Institute. And so there's behavior training on a cluster and the animal had transitioned to first the 2-photon rig and now the neuropixel rig as well. The idea in this behavior is to have an animal detect an image, and when the image is changed, to record response. So they are expected to lick a spout when the image is changing in front of their eye. And we monitor the licking response, as you can see on this image. And we have consistent behavior with this platform. We also have an associated publication. This was put together by Peter Gorbeski and Doug, Marina, and Sean. And we hope that in the next application cycle, we'll be able to sort of share it with you. All of this sort of platform, we've used them to make open data set. So the first data release was done a couple of years ago on the visual coding project. So this is the passively viewing experiment. And we made the same with the NeuroPixel capabilities. And so there's a large number of neurons across a number of recording sessions. And recently, we also introduced the visual behavior dataset with 2-photon recording as well. And so we have neurons doing this detection of change task. And so this dataset are sort of data releases, and they form the basis of what we can call surveys. Similar to how in astronomy, there's like really impactful surveys of the sky that are made available to the community. We think this forms the basis of an observatory model. And so I'm actually we were quite impressed when we started to look at the sort of impact in terms of publication of this platform. So here you see the different data releases from our end-to-end pipeline, what we've done in the last few years, and how the publication, either from internal work or external work, is ramping up. And I hope that you can see the symmetry with the similar publication figure I show you for the observatory. And so our goal with the OpenScope platform is to sort of make this available to the community so that we can sort of fulfill that vision. And so I'm going to hand this off to Carly, who's going to describe to you this opportunity so that you can sort of fine-tune your proposals.
Thank you, Jerome. So I'm going to dig into the details of this year's opportunity in the 2021 request for proposals. This is currently posted on our website for the OpenScope at the Allen Institute. So I encourage you to go and read it in detail if you have not already done that. Just another reminder, as Jerome said, we have done something similar in the past with Allen Institute funds, internally funded previous projects that kind of piloted the OpenScope potential projects from the community. And you can see both in 2018 and 19 lists of these projects that were done, and look at this for examples of what was done in the past. We are working now to expand on this with the NIH funding. And again, as Jerome said, this first year will be a specific solicitation with specific technical capabilities we are allowing. And then in future years, the technical capabilities may expand a bit and take on more complexity. But we do encourage you to look and pay attention to what the technical capabilities are this year and whether or not your planned experiment might make more sense for future years if more is allowed then. So again, just want to reiterate throughout this, pay attention to what technical capabilities are allowed for proposals in this first solicitation cycle. We'll be accepting 3 proposals this year for both that passive visual viewing of visual stimuli in both the ePhys with the neuropixels and the OPhys with a 2-photon. This will get more complex in the future years. As Jerome said again, we are leveraging that existing Allen Observatory pipeline, and we are specifying specific technical capabilities for this RFP in this round, either with the neuropixels or the 2-photon. And just a reminder, you can propose to do an experiment with either the 2-photon or the neuropixel, but not both. So to get into more details of the technical capabilities allowed for this round, you can look at the details of the RFP, and for example, in the 2-photon, starting from the transgenic mice, you can use and leverage existing Allen lines that are already established at the Allen Institute. These are listed in an appendix at the RFP. And we also specify what age range they will be at the time of the experiment, so 70 to 120 days old. Then when you move to the surgery, we are doing and allowing for proposals that have a 5mm craniotomy. And then it moves to the ISI mapping, where we'll be mapping major cortical visual areas. And then we specify a 2-week habituation period after that time. When you get to the 2-photon imaging, for this round, we are allowing experiments that do awake passive viewing of visual stimuli for the mice. Later years might have behavior involved, but for this year, it's just awake passively viewing. This can be done in 6 visual regions that are listed in the RFP, or something within that 5mm window. So make sure your experiment specifies these areas. This will lead to data from a maximum of 10 mice, quality control data, and the sessions can be 70-minute sessions for a grand total of about 90 sessions max is what you can expect to get data from. So you can have multiple sessions per mice. This is not the total mice we start with, but you expect to end with the 10 mice with quality controlled data. Similarly, for the neuropixels, if you look at the RFP, what we're allowing for this time, same thing with transgenic mouse lines, make sure the existing lines at that age, 5mm craniotomy, 2-week habituation. And then for the neuropixels, we'll have 6 neuropixel probes. Similarly, we define which visual areas you can use. It might be possible to get recordings from additional areas, but that should not be the core of your proposal. Again, you'll get data from a maximum of 10 mice, and you can have 1 session per mouse. The sessions are 120 minutes sessions, and so it'll be 10 sessions max, because for the neuropixels, it is 1 session max per mouse. So again, really encourage you to look at those details in the RFP for the technical capabilities and ask us questions and reach out to us if you don't understand those or have more specific details you need. For this round of the proposals selection process, it's a 2-stage review, and first there's a letter of intent, and then we will invite 15 of those letters of intent to submit a full proposal. The letter of intent is due September 22nd. It's 2 pages long. That will first go to a kind of internal compliance check within the Allen Institute to make sure it meets the instructions of the RFP and make sure it first and foremost meets those technical capabilities we list and is feasible within our existing pipeline. So again, just stressing, make sure you meet those technical capabilities first. Those that do will be passed on to that external scientific merit review, where we have people outside the Allen Institute looking at the scientific impact of your proposed study, and this will be a double-blinded process. From there, we're going to choose 15 of the LOIs, letters of intent, to invite for a full proposal, and you'll have about 6 weeks to then, from the invitation, to prepare that full proposal for a due date on November 22nd. That full proposal will be 6 pages long. That will again go through a similar process of a kind of internal compliance check to make sure it meets the instructions in the RFP and meets the technical capabilities and feasibility for the Allen Institute. And then it will go through the external scientific merit review to judge it by the scientific impact and your work plan to address the hypothesis you proposed. That will also be double-blinded, so make sure it's open and just judged on the scientific merit alone. From there, we're going to award 3 projects for this year's cycle, and those would start probably January 2022 to start the work. All of this is going to be done through an online platform for submissions called Submittable, and a link to that is on the OpenScope website where the RFP is posted. If you have any questions throughout this process of where you upload the PDF of your LOI and full proposal, you can always reach out to us at openscope@alleninstitute.org. If you are awarded, then once the award starts, here's the projected timeline is also listed in the RFP. We'll start with the awarded teams interacting with the Allen Institute staff to have a virtual project kickoff meeting. The awarded team will sign a research or collaboration agreement with the Allen Institute, and then you'll have discussions with the Allen Institute staff to select your transgenic mouse lines. And we also expect you to give the Allen Institute an outline of any potential details for initial pilot experiments to make sure this is going to work well and all the details are working well before the full experiment is run. So from early in the award, the Allen Institute will work with you and have a lot of back and forth discussion to do a pilot project, which is basically making sure everything is working well before committing to the full project. And then we want to have an updated experimental design if needed and any simulation code for the final data collection. Once all those everything's tweaked to make sure it's running well, we'll start the full data collection on the actual experiment. During that time, we expect one member of your team to come to the Allen Institute for 1 week to shadow data collection to see how everything runs. And we will have reimbursed travel for that. At the end of this period, you will receive, the Allen Institute will give you a production of NWB data sets, the neurodata without border data sets of your experiment. They'll be shared with your team through an online data repository like DANDI. And that will be exclusively available for you or embargoed for the first year unless you request it to be shared earlier. Once the Allen Institute hands off that data to you, your team will do the execute on the data analysis. And you'll have exclusive rights to that data in DANDI for the first year. After your data analysis, we then are looking for you to submit back to us a report outlining the results of your analysis, and also for you to pursue meet our open science mission of publishing the data in peer-reviewed journals, possibly in collaboration with the Allen Institute. So I just want to reiterate again if you win this opportunity, what you will get, your team will receive, and then what we expect from you in return. So each awarded team will receive access to the Allen Brain Observatory pipeline and the expertise of the Allen Institute. If you're proposing a 2-photon project, that means you will get these NWB files of up to 90 curated, or in other words, passing quality control sessions across a maximum of 10 mice. If you're proposing a neuropixel experiment, it will be NWB files for up to 10 curated, again, passing QC, single session across a maximum of 10 mice. You will also get that 1 week reimbursed visit to the Allen Institute to visit the Allen Brain Observatory for one team member at some point in the data collection. And then you will have that exclusive access or that embargo on NWB files on DANDI for 1 year after data collection ends. In the future, we're also looking at providing essential compute resources on Jupyter Hub server. And in general, this will get you access to what we hope to be a growing OpenScope community and just more interaction with the Allen Institute and others as well. What we expect then for this opportunity is one, amazing proposal. We're looking for impactful science and more hypothesis driven from the community, and see what you can suggest running and what you have, what you've proposed. That being said, it needs to be a feasible project with the simplest experiment design possible when using the resources we have at the Allen Institute, and it needs to align with the RFP and meet those experimental capabilities in the Allen Brain Observatory. So first and foremost, make sure you're meeting the constraints of the RFP, and then within that, trying to propose the most impactful science. We want teams committed to the project. So Allen Institute will be doing a lot of this work for you for the data collection, but we want someone who's around to engage with the Allen Institute staff and committed to analyzing the data once they received it. We're looking for teams to provide functional visual stimulus code and also interaction with the Allen Institute staff throughout any initial kind of pilot experiments or making sure everything's working well. Once you receive the data, we expect a team to execute on the data analysis plan and then also submit back to us a written report outlining the results of their analysis. We also have a mission of open science at the Allen Institute and looking for teams that are going to make efforts toward publishing either on bioRxiv and then in peer-reviewed scientific journals. We also expect we're trying to grow a community, so we want someone interested in engaging with the OpenScope community members and helping us build that community. And then agreement to disseminate results in a public release of data analysis and also in publications. So that's this year's solicitation cycle and what we expect in the RFP. If you have more questions, again, we have those individual consultations for 10-minute slots, and to look at the RFP carefully and reach out to us at OpenScope at alleninstitute.org for more questions. And now Jerome's going to talk about what the future solicitation cycles and the future of OpenScope could look like.
Thanks, Carly. So obviously, there's a large space of sort of experimenting system neuroscience, and each of these need special capabilities. And so we've put together a sort of a plan to try to sort of broaden the capabilities of the platform. And I'm going to talk about this in the next few slides. And so obviously, this is sort of going forward, and so some of this component could change in the next few years, but this is our current thinking. And so obviously, if you have sort of a very precise experiment that is not sort of you're not able to be able to do with this year cycle. You might be able to do this in the next few years. This is the goal of this set of slides. So we have 4 cycles in the next 5 years. It's a yearly cycle. And so our goal is in next year is to enable behavior. So we start with NeuroPixel and 2-photon multiplane imaging. And we'll start with the variance of the detection of change tasks, so the tasks that we know well. In future years, we want to sort of enable more flexibility on the behavior side. And so we'll develop variants of that behavior and potentially go towards a more general go, no go task. Obviously, the space of behavior is huge, and we are serving 14 projects. So we won't be able to address everything, but we'll try to go towards a more general go, no go task. And one other aspect is to introduce auditory cues, because I think it's going to enable a lot of different questions. Another development is related to a wider surgical access. I'm going to talk about that in the next slide. Okay, so this development was broken down into sort of 5 threads. The first one is about sort of the behavior extension. And we currently have in-house software to control behavior, but our goal is to adapt the standard that is currently used across the field. And so we are currently working to integrate Bonsai, which is a software was developed by Gonzalo Lopez and his lab at Champalimo and at UCL. The lab he was working as a postdoc. And so we are currently sort of integrating that software. So it's this thread here. And over the course of the few next application cycle, we'll first test its integration in our cluster, and then release it hopefully in the next application cycle, and sort of enable larger and larger capabilities. Our goal is to go towards sort of experimental proposal as software deployment. And so we control the hardware, and you will control the software. So that's the first thread that we are working on right now. The second one is about class storage. And so obviously we want to build a community around our data set. And so to enable this, we have to sort of make the data available. And so the first thing we are working right now is to standardize really well the conversion to NWB files and push this through the DANDI archive that we're going to hear about today. So that's why we have this key members present. And then, so once we have done sort of this data deployment, we're going to shift our resources to help develop a cloud analysis environment. So that will be a Jupyter Hub-based system where the team can log in and use compute resources to analyze the data directly on the cloud. So the data does not need to move anymore. The DANDI team has already made some headway into that, and so I'm hoping we're going to talk about this. So our hope is that by the end of year 3, we have a sort of a little more sophisticated cloud capabilities that enable teams to share code between each other, share routines, and build sort of a little more integrated ecosystem of analysis. The third thread is about, the third and fourth thread are related. They're about surgical access and recording access. So we want to enable NeuroPixel recording beyond the visual system. So it's already quite amazing to be able to have 6 probes located in the visual cortex, but we want to be able to have some of these probes relocated either further away in the dorsal surface or to record from a subcortical structure. And so right now we are doing a lot of development to sort of provide more flexibility in the NeuroPixel recording capabilities. And we hope to sort of release this in further application cycle, as you can see on this slide. Similarly, we're doing an effort on 2-photon calcium imaging to enable more flexibility in where we're recording from. This likely going to be still dorsal, so it's still sort of on the dorsal cortex in the mouse. But we hope to be able to enable recording in the frontal areas and potentially in the auditory areas, somatosensory cortex, so to fulfill more project about multisensory integrations and so on and so forth. We envision that the effort will take 1 year more than the NeuroPixel effort. So right now we are testing different approaches, including crystal skull-like approaches or moving the small window around. The last thread about technical development is related to the web portal. And so it's important to us that we're sort of transparent. And so the web portal that currently is hosting the RFP and the application portal, we hope to sort of release more description of the project and sort of have a miniature description of the analysis that was done by the different teams, so to sort of communicate about the life of the OpenScope resource. And so this will be an ongoing resource that's going to grow over the years to come. So these are the different development activities that we have ongoing. And so to summarize, we anticipate that in the coming years, you'll be able to have extended neuropixel recording location. So there will be subcortical and non-visual cortical areas. The details of these areas will be worked out by the work that we're doing now to figure out the efficiency and the yield of different recording capabilities. Similarly, for 2-photon, we'll be able to record from more widespread dorsal cortical areas, and so that will be made available in future RFPs. And the third and very important thread is about behavior. So we are currently working to deploy Bonsai to the platform. In future years, we'll have detection of change behaviors, potentially auditory stimuli cues inserted into the system, and have a couple of behavior extensions that will be made available to the community. And the last but important one is about data analysis. So through integration with DANDI Hub, the PyNWB that you're going to hear shortly about, the in-house LN SDK package and Jupyter Hub system, we hope to help develop a data analysis ecosystem around the project. So I've talked about the next 5 years, but obviously the vision is to build a sustainable future for observatory and system neuroscience, and this is dear to me. I think the convergence of science, community, and funding should enable sort of a 10-year cycle where first the community with the neurotechnology community identify a key technology that is helpful to the system neuroscience field, and then we build this platform for broad access to everyone, and go through this sort of cycle where the platform is used, and then the data is continuously deployed to the cloud for use by the theoretical community and sort of throughout the years. And at the end of the pipeline, we need to think about the people that are sort of dedicating their sort of professional life to this platform. And so that this way we take care of everyone. The experimental community can use, as well as the theoretical neuroscience community can sort of contribute and use the data and sort of test their hypothesis using this data set. And importantly, the neurotechnological community can contribute new technology that are disseminated very broadly. And so this is one of the hope for the future of sort of observatory in system neuroscience. So to summarize, I hope that we've convinced you that this is a new and exciting model for knowledge generation in system neuroscience. We discussed details of this RFP, and I hope you have some clarity about what to expect in the future years. So we'll have some minutes for Q&A now. Obviously, if you want to have a one-on-one follow-up, please reach out to us at this email address. We'll try our best to find some time to talk with you guys. Before I close, I do want to have the opportunity to thank everyone that contributes to this project. So the OpenScope is followed by a steering committee. So the community is vested in making sure that this resources is fulfilling your needs. And so these individuals here, we are very thankful for their contribution. Joel, Satra, Mackenzie, Adrian, Conrad, Nick, and Natalia, and Mariam Gale, they're all helping shape the project as the years to come. And so we have some ability to include your feedback and your preference. And we'll be using that opportunity to sort of move the project in the right direction. And also we are tremendously thankful for the Brain Initiative for funding this resource. And so it's really a community effort. And last but not least, there's a lot of people behind the scenes that are making this possible. So the scientists that validated and were vested in the success of the observatory, all of the research associate that was sort of running this experiment for you all. And so we're really thankful for them. There's each individual part of the pipeline is led by managers that are making sure that everything is working well. And so this is really important to success. And all of this is built by really talented hardware and software engineers that are making sure that this is working really, really, really well. And last but not least, the animals are taken care by the animal care teams, and we're making sure that this is done in the respects of their health and safety. And eventually, I want to thank our founders and the Yale Institute for supporting this work. So now we'll take some time for Q&A. Thank you.
Thank you, Jerome. So yeah, we are going to move to a Q&A section now. And just a reminder as well, if you can enter any questions, written questions in the Q&A chat box, Q&A box, not the chat box. So enter in the Q&A. And then if you have additional questions beyond this, we have those individual consultations that you should have received a Calendly invite for, and you can schedule a 10-minute slot to reach out to us for one-on-one conversations after this. If general questions on OpenScope or the specific request for proposals, and you can always reach out to us at openscope@alleninstitute.org, and we encourage you to do that. And please ask questions in advance with the RFP. We want to make sure you guys understand it well to get the best quality proposals and to meet those technical capabilities. I'm going to go ahead and the PowerPoint. And now let's see what questions we have coming in. Great. So I've seen a few questions come in so far. Okay. First one, Jerome. Will we be able to adjust the work with younger mice? So P20s to 30s, for example. And that 2-week habituation possibly disrupts the possibility to study development. So what about different ages in development?
Yeah, so I think in the RFP we listed the years. So we have to do the surgery at a certain age so that the skull is fully formed, because we have a protocol done at that age. So doing that in earlier mice will disrupt our current procedure. The habituation is important to sort of make sure that the mice are well habituated to the stimulus and to head fixation. And so within this constraint, we'll have to follow that. And so perhaps the passively viewing habituation could be maybe shortened slightly, but I think we should be careful with our current procedure.
So it sounds like, Jerome, for now, assuming the age range is only listed in the RFP for this round, and realizing that there's problems with the surgery at younger ages that we haven't tested out yet.
Yes, that's the main comment. Okay,
great to know. Another question that came in was, you mentioned choosing transgenic lines for NeuroPixel proposals, but what capabilities are available on the NeuroPixel platform that leverage transgenic lines? So how are you leveraging transgenic lines for NeuroPixels?
Yeah, so currently there's C57 mice as well as mice that express opsins, so that we can locate inhibitory neurons. I think that's the main capability with transgenic mice. We are currently not planning to have more transgenic mice in the NeuroPixel pipeline, but it's good to know if there's a lot of interest in integrating new transgenic mice. We could look into that in future years, probably.
And are there any past studies or something, for example, that leverage that for NeuroPixels that would be good to reference as an example? Think off the top of your head.
Our Nature, the publication that we put out on the NeuroPixel platform is a good reference.
Great. Okay. Another question coming in, are pharmacological interventions allowed for the experimental designs?
Not currently, no. The problem with pharmacology is all the dosage, you have to do it right. And so that's a lot of effort. So currently we're focusing on sort of visual stimuli modification this year. Pharmacology implies a lot of tests before.
And that might be a question mark for future solicitations as well. Is that right, Jerome?
The first year is likely going to be very constrained to all of this explorations.
Right. Another question, are optogenetic interventions possible?
Currently, there's tagging, opto-tagging. So that's what we can do currently. So you can see inhibitory neurons, locate inhibitory neurons in NeuroPixel. We don't have an optogenetic pipeline beyond that.
And this next question might follow from the pharmacology and the awake mice stipulation. Is anesthesia as an experimental condition possible? If so, what kind?
Yeah, we haven't done that too much. I mean, I could see isoflurane being a possibility. I will have to double check before being able to give an answer to that.
And that's something, I believe we have the names here, but you can always reach out to us by email if you want to get follow up on that question, and we can see if we can follow up with people as well. Okay, another question, are gene expression studies possible?
So that would be to locate, with like take the tissue out, and I'm not sure exactly what that meant.
This might be a better question for some back and forth one-on-one slots. So this is something might need bigger discussion of what exactly, or if you want to follow up with the Q&A box for more details on what you're looking for with the gene expression. But this is a good one to reach out to the OpenScope at lsu.org so we can have a more back and forth discussion on that. Okay. Another question. So talking about when we call that a pilot experiment to start in the initial, what do you mean by a pilot experiment versus the full experiment? Is this somewhere to try out things you haven't worked out yet or to get more data? So how do you envision that?
Yeah, so you have to think that an observatory is a different beast than and sort of a capability in experimental lab. So what we want is sort of give you access to the capability here and be confident that we're going to be able to sort of scale this up to a production level. And so the goal of the pilot is to figure out all of the technical details that are needed for the experiment to be a success. So you're going to deploy new stimuli. We need to make sure that this is working as you expected. And during the pilot, we might actually change parameters on the fly so to make sure that it's working. And so it's not going to be sort of being able to test a lot of responses of neurons to different stimuli. The goal is to sort of make sure that this is working. And so it's a lot about the technical test.
Most of the details should be sorted out in the proposal, and then it's just working on the, right?
Yes.
Great. Along with that, you mentioned in the NeuroPixels multiple different regions beyond the visual cortical areas. Can you clarify that a bit more of what's possible in those other regions?
Yeah, so currently we have 6 probes that are implanted at a precise location, and so that is currently fixed. And so that gives you access to all of the visual areas of the dorsal cortex, V1, LM, AL, PM, all of these. And there are some probes that are sort of going through LGN, LP, so that's subcortical visual structure, as well as the hippocampus, a couple of areas in the hippocampus. Beyond that, the subcortical structure, obviously as the probe goes deeper, the precision in the location is less accurate. And so that would be for later years where we have a little more fine-tuned abilities on the probe location.
Great. One more question that we have, does the current setup include eye tracking capabilities?
Yes, so we collect the data and we have this data set available to you all.
And then follow up with that, will eye tracking information be acquired in parallel with the EPHIS-OPHIS as in the current data sets?
Yes.
Okay, simple answer. Great. And then can you talk about, when we talk about getting 10 mice data from 10 mice max, does that mean the total you start with? Is that how many you're getting in the end? What do those numbers look like?
So it's important to keep in mind that what we're talking about with 10 mice is it's mice that went through our data curation. And so we're likely going to do more mice than that. So that means that this takes into account a failure rate, and sort of at the end, the mice that are collected are high quality.
Great. We had one more that you might have answered in writing, but I'll just repeat it for everyone. Is the OpenScope resource available to BSL2 labs and mice or samples?
Sorry, can you repeat that question?
Is the OpenScope resource available to BSL2 labs or BSL2 mice or samples?
Yeah, so I think it's related to the transgenic question. So this year, the particular transgenic line that we use is listed in our RFP, and so we are sort of stay within that space. The reason is so any mice that is running on pipeline goes through like a health and transgenics check. And so we experience that introducing new transgenic line can yield some problems. And so before we can introduce new transgenic lines, we need to sort of put a system in place. I think that's related to the question.
Yeah. We have another one on eye tracking available. So does eye tracking include pupil dilation?
Yes.
Okay. Another one for the multiplane. Why 8 planes 10 Hz for the multiplane recording?
So that's how the platform is set up. So the system is standardized. So we have an objective that is a large objective, and you can position this plane around. And there's a lot of sort of a downstream process. So the data is processed by the pipeline and then will be converted to NWB files. And so changing too many parameters will make us sort of have to change this entire process. And so we want to be able to share with you those files quickly. That's the reason for the constraint.
Great. And here's one. Which specific open licenses will data be released under? Is it the standard Allen Institute licenses, which forbids commercial use without special permission, or a custom license?
Yeah, I will need to double check on that because I think our plan is to release it through DANDI. We haven't done this process before, and so I'll need to double check.
Yeah, I think we'll check with legal team and we can have a conversation with that if people want to reach out.
I mean, the goal is to make it as open as possible, but we need to double check.
Another question, would it be possible to subject mice to specific habituation protocols, for example, visual stimulus conditions for adaptation?
We've done that before to have a sort of a different habituation. I mean, that's part of the design, right? So when you present new visual stimulus to the mice, they might need to be habituated to it. So I think it's likely possible.
And then someone's asking, did I stand it right that no optogenetic activation is available? Will any CNO experiments be possible if I want to change the activity of deep structures and record in V1?
No, currently that's not possible.
Okay. Is that something in future solicitations or just this round, or we don't know yet?
It's all of this will need technical development. And so currently we're not planning to develop optogenetic activation of subcortical areas. But if there's a big ask, that we might consider it.
And then bigger picture questions, and I think these two are kind of related, is why the focus on visual cortex and why inhibitory neurons, and just questions about the focus on the mouse visual system in general.
That's where we started the Brain Observatory. So obviously there might other questions in our overall sensory system, but we have the system in place for the visual system. So we're working to extend recording beyond it. So I'd mentioned this earlier in the talk, but that's where we started from. Obviously, if you want to have complete freedom, you need to build the system from the start with that freedom in mind. So it's a lot of effort.
Yeah, great. And in general, I think somebody's asking about having opportunity for additional questions, say the individual consultations. We are still scheduling those if you did request them through the initial registration. So you should have gotten a Calendly link. If you did not sign up for the individual one-on-ones during the registration, you can reach out to it by email. We had a lot of interest, so we'll see what time is available. We can see if we can slip in additional time slots, but you can always just reach out to us at the OpenScope@alleninstitute.org for additional questions. We want to make sure everyone understands the RFP as best as possible to get the best quality LOIs, letter of intents coming in. So please reach out to us if you have additional questions. Jerome, was there anything else you wanted to add and expand upon from the talk or from follow-up from the questions that were asked?
No, I think it was good. A lot of really good questions. So we'll have opportunity for more Q&A at the end of the webinar.
Yeah. So yeah, we will have an additional time if you guys have more thoughts throughout the talk at 11:30 for general Q&A. The next part, we're going to move on to talks by other collaborators in the community that I think are good for the neuroscience community to know about in general, but also will be important for the execution of these projects through OpenScope. And that's both the DANDI for the data repository by Satya Ghosh is going to be next. And then later we'll have a talk on Neuro Data Without Borders by Oliver Rubel. So I'm going to hand it off to Satya Ghosh if he wants to go ahead and open it up and share his slides.
Thanks, Carly. I will share my screen. Let me know if you can see the slides.
We can see your slides now.
Yes.
Wonderful. Should I get started, or do you want me to wait a minute or two?
Let's pause for 1 minute if people were joining just in time for yours. And I do want to just remind everyone, while we'll give them another minute before you talk your slide, in case anyone was joining right at 10, that this webinar will be recorded and will be posted on YouTube if you guys want to go back and see the details again and review any of the information we've given so far. So the whole webinar will be there if you want to look back at it, or if you missed the webinar or have collaborators that missed the webinar, you can refer them to the YouTube. And again, if you have additional questions from the first segment or these other ones, you can ask them at the end of today's at 11:30. We will have Q&A after each of the DANDI and NWB talks, and we'll ask that you keep those questions to the topic of DANDI and NWB. And if you have questions on, say, the RFP, we can save those for the very end when we're asking general Q&A, or save those for individual consultations. So I think we're close enough to the hour. So if Satya, you want to go ahead and get started.
Wonderful. Thank you, Carly and Jerome and the OpenScope team for inviting DANDI to this opening session for disseminating information about the RFP. We're delighted to be kind of part of this neuroscience ecosystem that OpenScope is producing, and I will cover a couple of things from the DANDI side to help you understand what DANDI is, and some details in some places, and I'd be happy to take on any questions in the Q&A section. So for those who don't know, DANDI is a data archive, but we kind of think of it more than a data archive. We think of it as a collaboration space for neurophysiology projects, and hence our effort in this particular space with OpenScope to complement its kind of interests and wishes of creating an observatory fits in quite nicely. This is a project that's done, run out of MIT, Dartmouth, Kitware, and with Catalyst Neuro as partners. And I'll tell a little bit about the team. Yes, so as I mentioned, we have folks at MIT, Dartmouth College, and Kitware and Catalyst Neuro as partners in this project. We're supported by the BRAIN Initiative, and we're also strongly thankful to the AWS Public Dataset Program, which houses the data that's on DANDI. This would not also have been possible without strong collaborators, which includes the Neuro Data Without Borders, which you will hear about in a bit, the Brain Cell Data Center, which is part of the BRAIN Initiative Cell Census Network, the Allen Institute for Brain Science, the BIDS community, the OpenScope project that you're here for today, the International Neuroinformatics Coordinating Facilities, the International Brain Initiative, and various members of the community who've helped out in various ways in getting various pieces of data on DANDI online. So this won't come as a surprise to most of you over here. Neuroscience today is multi-scale and multi-species, and it's being driven by instrumentation, experimentation over different kinds of scales, over different types of in vivo and ex vivo work to help create new theories, new models and experimental designs, but it's also kind of being used to drive various biological applications, whether it's understanding certain types of disorders, whether it's observing behavior and knowing how to nudge behavior in a certain way, and more generally as a basic science in kind of creating knowledge that then helps drive biological applications. And alongside this very complex space, there's a lot of tools being created. These could be new instruments, new protocols, new analysis tools, and all of the work, the experimental work, is driving the development of a lot of data at all kinds of scales with rich descriptors and provenance. So there needs to be an infrastructure to kind of help all of this work that's going on that facilitates dissemination, search, visualization, and computation. And hopefully we can do this as a collaborative enterprise and coordinate across different kinds of projects that are going on and provide data with as much fairness and efficiency and analytics also through that same lens of fairness and efficiency as possible. So as an example, you saw a piece of this workflow in Jerome's talk. This is the Allen Brain Observatory NeuroPixels workflow. And today, while we think partly about the data that comes out at the end in the form of digital files, there's a whole process that's involved. And in this particular case, a key goal of the archive is to ensure that all of the details of this process are captured as much as possible to enhance the future reuse of this data. The NeuroPixels workflow results also in a scale of data that's growing by the day. This was originally developed earlier in this decade, and today we can capture a lot of data in a very short period of time. Now, this is talking about 2 simultaneous insertions. You heard about the OpenScope program having 6 insertions simultaneously. So this leads to a large amount of data that has to be captured, has to be processed, has to be understood in a way for future folks to kind of take on and do things with. And while the neuroscientific community might extract certain pre-processed sets of data to increase usability downstream, a lot of the processes in terms of what we should be doing with data are not set in stone yet, and hence having a place to put the raw data is going to be important across the different kinds of experimental techniques and modalities being used. So this is where DANDI kind of comes in. The DANDI archive is a Brain Initiative archive for publishing and sharing neurophysiology data, including electrophysiology, optophysiology, and behavioral time series, and images from immunostaining experiments, whether they come from microscopy or associated MR images. And I want to highlight one little term now. The word data set is too overloaded in the space of science. So we created a new term called the DANDI set, which is simply defined as an organized collection of assets. You can think of them as files with both file level and data set level metadata generated from an experiment or a project. And this archive primarily comprises of a few components. There's a web application and an API server. This is what you would mostly use to interact with the data in the archive. You'll be able to browse it. You'll be able to interact with it programmatically. And I'll go into a few more of these details a little later. Alongside this application and API server, we're using standardized descriptors that are being supported by Brain Initiative awards. 2 of these, or 3 of these, are the Neurodata Without Borders, the BIDS or Brain Imaging Data Structure, and the Neuroimaging Data Model. And these all help us kind of understand how to encode data to provide maximal reusability and maximal understandability of the data downstream from the person or the team that produced it. So hopefully with OpenScope adopting NWB and other components of this, it makes it more usable for others who are going to be looking at this data away from the team that produces it. There's also an analytics space that Jerome referred to. We're running a Jupyter Hub instance that you can use to play with data in DANDI. And this makes it quite nice because the data sits in the cloud. You get to do analytics on the cloud, which reduces the amount of data you need to move out of the cloud to do things with. And finally, we have a set of Python clients and other resources like DataLad to interact with the archive. Now, all of this uses standard files and formats, and we're working with the community where we don't have ideas of what formats or things to use, where we want your input to figure out the use cases that you have that would benefit from some of the advances in technology that we're working on. So I'm going to go through a few examples of data in DANDI just to illustrate the scope of the archive. Here's an example of a data set that was used by a software framework called Spike Interface and a paper that looked at what's the agreement across spike sorting tools in terms of detecting the units detected by each of those software. And this, as with many things in neuroscience, broadly speaking, each algorithm makes their own assumptions and hence results in different kinds of ideas about what should be extracted. This particular data set, alongside all the tools and the scripts that they use to analyze the data, are all available on DANDI. Another example is this microscopy data set. So this is a light sheet image of the human brain, and this is work in progress. Each slice is being collected, is being processed, stained, restained, data is being collected and uploaded to DANDI on an ongoing basis. At least for this one hemisphere of the human brain, we're looking at DANDI as a place that stores this particular DANDI set as almost like a Google map of the brain. So it's one single brain. Can we create a single architecture that stores information at the cellular level across this entire hemisphere that allows you to interact with it and work with the data in the cloud? So we are at about 14 terabytes of data for this brain, a few slices in right now, but this will continue to grow to be a bigger data set as more data stream in. And here's another example of a DANDI set that's being created that involves 4 collaborating sites, 2 at Boston, Mass General Hospital and Boston University, 1 in Italy in Florence, and 1 at Columbia. And they're using DANDI as a way to push different kinds of data into it. So the process here is a workflow that's not at a single institute. It starts with doing ex vivo brain imaging at MGH. It goes over to Boston University to do OCT, or optical coherence tomography, then goes over to Lens to do light sheet imaging with different kinds of antibody-based stains. And then finally, the digital data goes over to Columbia to try and do stereologic counting of cells in different parts of the brain. And this needed integration, it needed standardization, not everything was set in place, and DANDI is providing the resource for these groups to collaborate on a single data set that organizes these different modalities together and allows anybody in the world to access the data to further look at it. So we have built DANDI on top of open source technologies. Every aspect of DANDI is open. There's certain things we have done so far that allow us to deliver DANDI as a resource to you. This includes the web apps, the Jupyter Hub, and indexing all of the DANDI data through DataLad. There are other things that are kind of on their way. We're working with various neuroscientific communities on building converters to NWB. This is together with the NWB team and Catalyst Neuro. And we're trying to incorporate other kinds of technologies that would make data easier to sync across places into DANDI. And finally, there'll be a few things that are coming in the pipeline that I'll talk about a little later, which has to do with processing data as it hits DANDI and ways to interact with the archive through better means than just programmatic clients or the browser. You can find the web portal and the analytics things all on DANDI. There's a GitHub organization, which we also use as a help desk for DANDI. You can contact us through help@dandiarchive.org. And for any other questions, come to the help desk. Our intent of building this on an open source stack is to create a set of things that's accessible to all kinds of people, whether they be hardware or software developers, the scientists, the data contributor, or citizen scientists. DANDI as an archive is accessible to high school students, college students, or anyone who has an interest in science to be able to play with data online without having to do collection. And anybody who builds or provides data towards DANDI is helping increase the diversity of this ecosystem. So let's kind of take a look at the DANDI ecosystem a little bit further. We have a few components, and over the next few slides, I'm going to go over some of these components. The entire archive sits on AWS, and you can interact with it through various tools that are not just in DANDI, but also external tools like the NWB Explorer from Open Source Brain, or the Bioimage Suite from another Brain Initiative project that allows you to view data that's sitting on DANDI. The heart of the archive is an API server. Now, this might not be relevant to most scientists out there, but anybody who's in the team trying to talk to DANDI will want to know what the API server does, because this is not just for pushing data into DANDI, it's also being used for pulling data out of DANDI. And so if you are going to build your own tools and interfaces to DANDI, the API server really implements all the archive metadata and data logistics. We're uploading data to a public S3 bucket, which is provided by the Amazon Public Dataset Service. And we deduplicate data, which means if you upload the same data from different places multiple times, there's only one single copy of it left there. It also allows us to publish DANDI sets, and I'll get into the publishing component in a bit. This interface is public, but editing operations, so anywhere you write to the archive requires authentication. This is the main web UI, the DANDI archive, and it's what most people use to interact or browse things on DANDI. It talks to the API service to create, browse, search, and publish DANDI sets. You can view data sets and assets metadata. You can download individual files, at least for small files. You can just click on it and download it. But for bigger DANDI sets, we would want you to use a client library we have built to move the data, or just use the hub to access the data. The dataset metadata records can be edited using this archive, and that's what enriches each DANDI set to be more useful for downstream consumption by others. Now, most of DANDI could have been just a Google Drive or a Dropbox. People upload data and store in some of these places all the time, but just storing data on its own is insufficient. You need to be able to use the data, and that means understanding the data. Sometimes people will say a README is sufficient, but we think in the 21st century, we should have data curated with more than just a README. It should be structured, and it should allow for both human and programmatic exploration of the different kinds of data that are there. So we are using Neurodata Without Borders, BIDS, and NIDM incorporated into a metadata model for our DANDI sets and assets. And this is accessible for many different people through different kinds of data formats that they can use programmatically, as well as through the visual interface on the archive that you can use to browse and search for information in these DANDI sets. For those who are uploading data, there's an entire workflow in place. For this particular program in OpenScope, most of the data upload will happen initially by the OpenScope team, but you might imagine that other kinds of derivatives would be uploaded by the people analyzing the data. And so both sides would need to understand how to upload data into DANDI, but more generally, this is how we kind of create a framework where we crowdsource the creation of the data set at the place where people have most knowledge of the data. So we want you to validate your data on site at the client side. We want you to organize it on site at the client side, and we provide tools to help you to do so. And then you upload it to DANDI. So once you upload it to DANDI, it's already organized, it's already standardized, and you can yourself use it for doing research. So DANDI should not be seen as an endpoint for publishing data, but as a place where you can play with data and you can work on your own research as you produce the data. And finally, there's a publish side to DANDI. We kind of think of DANDI as having draft data sets. These are data sets that you've uploaded, you're still playing with, but whenever you're ready, you can hit the publish button and mint a DOI for the data set. And this allows you to cite this data set as any other research or scholarly output out there. Finally, we are also incorporating DataLad, which is a tool for organizing or accessing data with rich version control as you work on data. So this could be about collecting data, it could be about transforming data through various applications, and every DANDI set is accessible as a DataLad data set as well. The data resides on S3. You clone, just like you might clone software from GitHub, a DANDI set locally, which does not transfer all the data to you, but transforms the structure of the DANDI set to you. And then you can retrieve any piece of data as you want to play with it, view it, work on it, etc. For more details, you can look at handbook.datalad.org, how DataLad is useful not just in the DANDI context, but how it could be used in the more general scientific context for you to create precisely versioned copies of your data. So I'm going to end with kind of discussing a few of these upcoming features and the technical roadmap. We're continuing to make user interface and user experience improvements. As you work with the archive, if you find something that you're not comfortable with, you find something annoying, please reach out to us through the help desk, and we'll take all comments seriously and into account. We're increasing the hub to integrate more applications. So things like spike sorting and cell counting, other external services and applications like the Raves suite for intracortical recordings, and GPU-based containers like Kilosort and other CNN or DNN, deep neural network-based applications. So the hub becomes a place where we can play with a lot of tools and see the interaction with the data. As I mentioned earlier, one of the key challenges for many labs is how to convert data to NW. In the context of the OpenScope project, the Allen Institute team, Mindscope team will be taking on this conversion process to create a consistent format to upload to DANDI. But we are happy to work with all the users out there to figure out better ways of converting data from their instruments into NWB together with folks from the NWB team. There's a lot of custom instruments in the neuroscience space, so everything does not easily flow as we would expect it to. We're going to continue to provide better metadata management and search. The key idea here is, let's say all of your data in your lab got hosed away because of data corruption, fire, etc. Would DANDI be sufficient? Would the data that you have uploaded to DANDI be sufficient for you to continue doing the kinds of science you're doing? Which means, have you provided enough metadata alongside the data to continue doing the science? And this is where we need better measures to help you provide such data. We need to work with the community to better understand what kind of metadata would be more useful. And then finally provide the search capabilities to search the data in effective ways so that you can see whether you actually need to acquire new data, or is there data already available that you could use for questions that you have. And towards this, we're kind of building a scientific front end that will link data to questions. As data are starting to come into DANDI, we're interested in seeing how much of this data can be reused for other kinds of questions than what the original lab thought was relevant to the data. And that's really the goal of a lot of these public archives, is the reuse capability for questions that the original data were not collected for. We're also working with various external services like Metacell, Neurocast, and DataJoin to figure out how the data in DANDI could be useful to researchers using those services, and how those services can link to data in DANDI. On the technical side, our focus this year is on improving search and the embargo process. Now, Jerome mentioned about the embargo process in the context of OpenScope. This feature should be available in the next month or two. So by the time your data gets collected, this would already be in place. Our next move on the DANDI side is really to move towards analytics a little bit. How can we decide what kind of process data to provide on top of these larger raw data sets that would be useful to the community? And for this, we need your help. We would like to better understand the kinds of tools and techniques that you use that would be useful to process data with as soon as the data hits our servers? And further, how do we work with you to figure out better ways of tracking provenance, both on the data collection side and the analytics side, that would be useful, again, to you as a downstream user of your own data or to others who are using your data? Beyond that, a lot of our features are focused on making DANDI distributed. The acronym DANDI stands for Distributed Archives for Neurophysiology Data Integration. And our dream vision is that every institution, every lab has kind of a copy of this DANDI infrastructure locally, because we expect the scales of data to grow significantly over the next decade. And these individual DANDI instances kind of talk to each other and talk to a central DANDI instance to move data around efficiently or move compute efficiently to the instance. Now, this is a vision we'll only touch elements of in our initial deployment cycle in 2023, but we're hoping that this becomes a federated space that connects different institutions and labs to each other to even further understand what data are being produced and who can use different kinds of data as effectively and efficiently as possible. Finally, there's a lot of Brain Initiative archives being created. We're hoping to achieve cross-archive interoperability, because these archives are being created with modalities in mind. So neurophysiology data comes to DANDI, transcriptomic data goes to the NEMO archive, other microscopy data goes to BIL, electron microscopy data goes to BOSDB. As neuroscientific experiments are getting more complex and complicated and across modalities, there's a need to kind of specialize the individual features of each archive, which are dedicated to each modality, but also link across them. So we're hoping to work with the other archives and with the NIH Brain Initiative Informatics program to try and develop cross-archive interoperability. So let me end on a couple of notes. We consider DANDI to be a FAIR archive. FAIR, for those of you who are not familiar with the term, stands for findable, accessible, interoperable, and reusable. How is DANDI findable? We're using a rich schema with use of standard ontologies. All elements, DANDI sets and assets, carry unique identifiers associated with the data. They're searchable on DANDI archive, and linked data is exposed for others to build search enterprise on top of DANDI. We're using standard file containers, whether it's Neurodata Without Borders, NIfTY, TIFF, HDF5, NGFF, which is a microscopy format that's being developed, and others. We're exposing metadata also in standard formats like JSON and JSON-LD alongside data. We're using standard protocols. There are no special components to create to talk to DANDI. And we are providing access through multiple routes: the web interface, the Jupyter Hub, the API, Python clients, DataLad, and working with other communities like the HDF5 community to extend streaming support to all the objects on DANDI. We're using standardization of metadata and data formats to provide interoperability. Now, creating different formats just because we can does not lead to greater reuse of data. As a community, we have to come together to understand the bottlenecks as well as the advantages of each format and work towards resolving those bottlenecks. Data are going to keep evolving. We as a community have to be able to kind of move the standardization of these formats both towards efficiency as well as maintainability. But we can't come up with a new format every year to help with this process. We're using standard schemas, whether they be identifiers.org or the W3C provenance model, and a whole host of other things to kind of encapsulate the metadata and dealing with things within the context of ecosystems, both the NWB and the BIDS ecosystems. Finally, everything in DANDI is reusable. The license is specified. There's a rich metadata schema, and all components of DANDI, not just the data sets, but all the software components are released under free and open source licenses. So I'm going to stop with this. DANDI is a FAIR archive for neurophysiology data, and it's a collaboration space for you. You can really use it from the point of data generation, and we encourage that. You can create a DANDI set without putting a byte of data into it, saying, I'm collecting this kind of data. with the hope that other people who are interested in that kind of data can start collaborating with you towards designing principles. And in some ways, that aspect of DANDI is very similar to the goals of the OpenScope program in creating a resource that's kind of generated by the community from the data collection space itself, not just post-publication. With that, I'll stop sharing, and I'm happy to take any questions.
Thank you, Satra. So that's a great introduction and overview. And if you guys do have any questions, please enter the questions in the Q&A box again and reach out. And it's a great overview just to know this resource is available to everyone outside of just OpenScope, but this is something we will be leveraging for OpenScope and how you will be accessing and receiving that data that results from that. So I want to start, Satra, is there other resources are coming, learning events, some kind of resources they can go to learn additional or just begin playing around with this so they don't even know where to start or just want to learn more about it?
So wonderful question. So there are a couple of things over the last 18 months since we've been virtual for almost everything. There's a lot of resources that just between the NWB team and the DANDI team we've put out online. So those could be great starting points to kind of look at. In terms of upcoming workshops, some of us, depending on the situation, will be available virtually during SFN as well as in Chicago. So you're welcome to kind of reach out to us. And we are planning a workshop in early spring. We were certainly hoping that we can have this workshop in person rather than virtually as we've had. And so there should be a workshop coming up in early spring for DANDI. But between now and spring is a lot of months, and people are welcome to reach us through the help desk, and we can arrange certain things. But I know you're doing consultations. If anybody is interested in learning more about DANDI, we'd be happy to chat with them on those fronts.
And that's a great reminder. If people are scheduling their consultations, you can put in a box of what type of questions you have. So if you let us know in advance what type of questions you have for one-on-ones, we can see if we can tie in someone else's thoughts with expertise in that and be ready and have resources for you to get more specific answers instead of just the Allen Institute staff. No, it's great to know events are coming up so they can help where to start, and hopefully there'll be some in-person ones soon. But it's nice the last point you had about this is a great resource, even if they don't have specific data they need to add there, but they can start adding the framework for others to use and take advantage of as well. Sounds like a great place to start playing with it. We did have a question here. Is it possible to access, open, and analyze data posted in DANDI with MATLAB?
The short answer is yes. Now I'll go into, I don't know if Oliver is going to cover a larger component of it, but NWB has accessibility both on the Python side as well as the MATLAB side. And one of the elements of this is, one piece we are missing right now is we don't provide MATLAB licenses in the Jupyter Hub that's online. And we've been talking to MathWorks about figuring out a mechanism to do that. But what you can do is in those contexts, download data and use your university's MATLAB license. Or in some cases, your university may allow you to create virtual private clouds, which allow you to access your university's MATLAB licenses in the cloud itself. And that would prevent kind of larger movements of data if you chose to avoid that. So it partly depends on which institution you're in, what the license settings are. But we should be coming to a point, hopefully with MathWorks, where there is some element of MATLAB computation available on the hub.
Great. I was wondering also, can you go into more about what type of raw data files can be there, or the limits on that, say videos, etc.? What can be added there and what are the limits?
Great question, Carly. As an archive or a storage space, you can pretty much upload anything to it. However, that's where we want to kind of create a difference between a Google Drive or a Dropbox and the archive, in the sense that we want information organized in a way that makes it useful to downstream use cases. So we're providing a minimal structure to each DANDI set that's kind of inspired by the BIDS ecosystem. Of saying there's some organization, most researchers can map onto that kind of organization, and we are going to name files and structure them in a particular way. Now, we're using NWB for neurophysiology data sets, and sometimes there might be videos associated with them. We're working with the NWB team to better figure out their external reference implementations. Now, this is a little bit of a technical detail, but something in that NWB file has to say that there is an external file that's associated with this time series. And that's work we're doing. From an archive side, the video should be uploadable as an MP4 or something alongside the NWB file. We're trying to figure out the details of making the connection between them inside the NWB file itself, such that it can be used both from a DANDI perspective, but also if you downloaded it locally, that somehow we can persist those links in some manner. For many things in electrophysiology, you would want to put it in the NWB file itself. But there are other things for which the community has created formats and standards that are highly efficient, movies being one of them. And for that, it does not make sense to kind of convert all the frames into NWB, although one could do so and store it inside. But that might be places where an external file might be useful.
Great. One other thing I wanted to clarify too is, can you talk more about the difference between kind of keeping the data for your own internal use privately while using DANDI versus the public? And you were saying that embargo is still being developed. So what is possible now versus the future, and where is that line?
So I will say right now, everything is open. So there is no private component to DANDI right this minute. If you were to come to DANDI today, everything you upload to DANDI is publicly viewable and downloadable. I mentioned embargo is in our about a 2-month horizon, I would say. We're talking to AWS about a few things, but hopefully we'll get that feature implemented in the next 2 months. What would happen then is that only the owners of the data set would be able to view and list the components of the data set. The metadata around the data set, as in here's a description of it, here's some keywords, we would still like to keep public, but we'll work with the community to better understand the embargo needs on that front. We do think that some minimal exposure of metadata to the outside community can be useful even for the people who want to keep an embargoed data set private, because that might bring in other kinds of collaboration. So we're also going to build a management portal that makes that component of it more like Google Drive, in the sense, if a reviewer wants to see your data before you release it to the world, they could kind of go and look at it. Or if you want to collaborate with someone and you wanted to add them not as owners, but as viewers of the data, they could kind of do so. So those are some of the features that will come alongside the embargo. But the idea behind embargo would be, it would be under control. Now, from an NIH perspective, they have a data sharing policy that we are going to follow. But if somebody wants to create a different policy that still broadly is in line with the NIH policy, we would be happy to kind of converse with them and talk about. I can say from what Jerome mentioned that that policy would be consistent with the NIH policy, so that wouldn't be different from the OpenScope program's current guidelines on data availability.
Great, thank you. Another question, could we store in vivo neurophysiological data?
Absolutely. There are many examples of it already on DANDI in all kinds of ways. So there's in vitro data, in vivo data that sits on DANDI, and you're welcome to store. The one place where we want to be careful is simulation data, because that can blow up the archive almost instantly overnight. So if you're planning to store simulation data, please reach out to us before you start uploading terabytes of simulation data.
And that leaves another question is, what's the best way to reach out to the team if you have specific questions or just want to know where to start? Is it the help desk? Is there a better resource?
The help desk is a good place to start. We'll monitor it. You can also reach out at help@dandiarchive.org.
And I'll just add, if anyone has questions as well and want to reach out through the OpenScope at alleninstitute.org, we can always forward it along if it's a DANDI specific question as well. Great. So it sounds like there's a lot of events coming up to learn this and start playing around. And even if you don't have a direct use for it now, it's worthwhile to go in and start playing with it and seeing what's feasible and setting up. And then a lot of interesting features coming up in the near future as well. So great. So we will be using this for OpenScope of how we hand off the data to the teams once we've collected it. And it will integrate nicely with the NWB, which we'll have a talk about next. And again, just reminding people, if they have more questions on this, to let us know before the one-on-ones or in an email so we can try and connect with you before talking to those potential proposal submitters, see if we can get more direct conversations going and have you there to answer the questions more specifically. Anything else you wanted to add, Satra, that we didn't get to talk about during the presentation?
I think the only thing I would add is this is a community resource, and in the spirit of things, as Jerome started off earlier, we want to make this ecosystem richer, nicer, and work with each other. So think of DANDI as something we implement, but something that you can guide. And so reach out to us to see and suggest things that we can do to improve the archive to help you do your scientific work.
Perfect, thank you. And again, we will have more time for questions at the end after the NWB talk as well for general questions. So if anyone hasn't thought of their question now, you can still ask it at the end of the day today as well. Great, well then I'm gonna pass it along to Oliver to talk about Neurodata Without Borders and introduce us to that as well. So if Oliver, you wanna go ahead and share your screen.
Will do, give me just 1 second.
Sure. And again, if anyone wants to look back at this conversation later, it will be recorded on YouTube and you can go back and refer to this if you didn't manage to catch anything then, and share it with your collaborators. So it will be available on YouTube for reference in the future. All right, and I can see your screen, Oliver.
Perfect. All right. Well, thank you everybody for joining this amazing webinar. My name is Oliver Rubel. I'm a staff scientist in the Machine Learning Analytics Group, as well as the Computational Bioscience Group at Lawrence Berkeley National Lab. And I've been involved in a leading role with the development of NWB since about late 2016. And so in this talk, I will give you an overview of NWB as well as the larger NWB neurophysiology data standardization ecosystem and discuss how NWB helps drive collaboration in neuroscience. One nice thing for you, if you're using OpenScope, is a lot of the details that sit under the hood of NWB, you probably don't need to know about because OpenScope nicely will convert all the data already for you into NWB. So you will only need to use the data, but it's still good to know, I think, about the inner details here, because like Satra mentioned, you will not just be consuming NWB data from the archive, but you will likely also generate new derived data products that you might need to share through the archive as well, and as such generate your own NWB files. And so knowing about how NWB is structured and how it works is really useful for that part as well. And so first I'm going to discuss some of the core challenges that motivated the creation of NWB and give you a high-level overview of what NWB actually is. And then I'm going to discuss some of the more core nitty-gritty details of the data standard ecosystem that NWB defines. And then at the end, I will discuss some of the energy barriers in data standardization that we face in the community and how we try to address them with NWB. And so, first of all, neurophysiology data are really expensive to collect. And at the same time, neuroscience data sets are very rich and can often be used to answer many different questions, even years after they have been acquired. And reusing data saves money and time, it reduces animal use, but at the same time, it is currently often very limited in its reuse because there's so many different ways in which people capture the data. And so that's why we're also so excited to have OpenScope work with us and the DANDI archive to really make the data that they're generating open and accessible. Second, sharing neurophysiology data within a lab and with collaborators is often very tedious. Everyone has different ways of storing the data, and metadata required for interpreting the data is often missing or stored ad hoc. And so that is where NWB can really help standardize what metadata is being required. And then also through all the mechanisms that Satra already outlined, where you're now able to find your data online and search collections of other people's data as well. And then finally, scientific results and analysis are hard to reproduce and compare. Processing, analyzing, and visualizing neurophysiology data requires converting data often from one esoteric format to another, while important metadata is often inconsistent or missing completely in this process. And so that is why we set out to create with NWB a new data standard for neurophysiology data and related metadata to really address the issue of making data shareable and interchangeable and reusable. So NWB then is an ecosystem for neuroscience data standardization. But what does it really mean? So first of all, NWB defines a unified data standard for neurophysiology data that is focused on the dynamics of a group of neurons measured under a large range of experimental conditions. NWB supports a broad range of data types from end-to-end neurophysiology experiments, which includes recording of neural activity via extracellular and intracellular electrophysiology, as well as optical physiology, behavioral data, as well as metadata about the acquisition systems, such as devices, experimental designs, such as stimuli and trial structures of experiments, and experimental subject. And NWB also supports the concept of formal extensions to the standards to enable us to add new data types and modalities, as well as lab-specific metadata or metadata about acquisition systems, processing, analysis, or experiments that may not be covered in the core standard. So you can create your own extensions to the standard to add new metadata or data into the file that is specifically required for your experiment. And so NWB enables unified storage of multimodal raw and processed data. So just as an example, here you can see a pipeline of an experiment that utilizes both extracellular electrophysiology and optical physiology to measure the neural activity. And using NWB, the scientists can store both the raw optical and the electrical recordings, as well as the subsequent primary data processing results from spike sorting and RI detection in a single file. And so with this, NWB provides a critical conduit to facilitate analysis and complete data from neurophysiology experiments. For example, the interactive dashboard that you see at the bottom of the slide shows on the left side the acquired image and the outline of a single segmented neuron in red. And then on the right side, you can see a juxtaposition of the extracellular electrophysiology signal, the extracted spike times, and the simultaneous fluorescence for the same segmented region, so that you can analyze all this data in a coherent frame and all from the same data standard. But NWB is more than just a file format. It really defines an ecosystem of tools, methods, and standards for storing, sharing, and analyzing complex neurophysiology data. And so here you can see an overview of the NWB software ecosystem. At the foundation, we have the NWB data standard schema, which defines the rules of how to organize experimental data and metadata, meaning which data is required, what should you call the data, where does it need to be placed, and so on. And then extensions to the NWB data standard are managed through our NeuroData extension catalog. And then using the PyNWB and MatNWB reference APIs allows users to easily create, read, and write and use NWB files while ensuring compliance with the NWB data standard. These tools also include additional tools such as tools for extending NWB, validating NWB files, and other essential functionality for using NWB files and interacting with the data. And then higher level tools, which are shown in red, facilitate conversion of data to NWB, introspection, exploration of NWB files, and creation and documentation of extensions, among others. And then finally, there is a growing collection of community software tools for data analysis, visualization, and management that use NWB technologies to provide additional rich functionality to end users. And so NWB technologies are really at the heart of the neurodata life cycles. Data standards are a critical conduit that facilitate the flow of data throughout the data lifecycle, from acquisition of the data to processing, analyzing, to publication, preservation, and reuse of data. And they facilitate integration of data and software across all these different phases of the data lifecycle. And so what this means is that NWB needs to support the needs of and integrate with technologies across all these different phases of your data lifecycle. And so our goal with NWB is really to work with, not to compete with, existing and emerging data technologies. And so a large part of what we do is really work with the community to make sure that the tools and methods that are most relevant to the community are supported through NWB. And then finally, NWB is a standard for, not a standard of neurophysiology experiments. What this means is that NWB does not aim to tell you how to perform your experiment or standardize experiments itself, but we aim to standardize how you generate and save the data that is generated by your experiments. And so here you have a quick and rough overview of some of the analysis tools that are currently available to you that directly support NWB. These are all tools that are being generated by the community to help the community analyze data. So at the first level, you start with PyNWB and MATNWB, which are our own reference APIs for NWB in Python and in MATLAB respectively. And then as you move up from there, other tools are building on top of PyNWB and MATNWB, such as NWB widgets or NWB Explorer, which are then general purpose tools to visualize and analyze NWB files. And then tools like Field Trip, Cell Explorer, Brainstorm, and Spike Interface provide advanced capabilities for processing and analyzing extracellular electrophysiology data. And then similarly for optical physiology data, you will find tools like K-man, Sweet2P, or calcium image analysis to process optical physiology data. And then DataJoint is an open source framework for programming scientific databases that is used to manage computational data pipelines as well as large collections of NWB files. And then finally, there's DANDI, which you already heard about, which we use as a reference archive for the NIH Brain Initiative. And as you already heard, NWB is one of the core standards that DANDI supports. And with the latest release of PyNWB, you can now also access your NWB files on DANDI in a streaming fashion, or really from any S3 bucket. Which what does it mean is that you can now open your file through the Python API while the file is stored on the S3 bucket, and you only stream the data to your local machine that you actually need for your analysis. And we expect more tools to become available in the near future. For example, OpenEFIS GUI, Deep LabCut, and Beetle, and open source Brain 2 are all working to add support for NWB already, and more tools are already in the pipeline as well. And so now that you have a high-level overview of some of the things that NWB can do for you, let's have a peek under the hood and take a look at some of the core technologies in the NWB data standard ecosystem that make NWB work. The goal here is to just give you a general understanding of how NWB works, so that if you have to generate your own data in NWB, you understand what pieces are relevant to you and how to use them. And so one of the key challenges in the development of experimental data standards like NWB is the diversity of stakeholders and the data challenges and requirements at the different phases of the data lifecycle, from acquisition to analysis to publication. And so a key aspect that has allowed us to manage these diverse requirements and discussions in the community is that we already early on in the process of developing NWB clearly outlined the main components that we need to make in order for the standard to work. And those components are outlined in that triangle figure that you can see on the slide. And so first we have the NWB specification language, which allows us to formally define how neuroscience data standards need to be organized. And then the data standard schema, which is shown in orange, uses that specification language to organize complex connections of neuroscience data. So this is probably the component that most of you will think of as the standard. And then the data storage component looks at how can we take all these objects that are specified in the standard and translate them efficiently to disk for storage. And then finally, the data APIs allow us to efficiently interact with the neuroscience data to read, write, and query and analyze NWB data. And so in the next few slides, I will go one by one through these different components to clarify a little bit more what they do. And so first up is the specification language. The role of the specification language is to allow us to formally define data objects like groups, data sets, links, and attributes, and their features such as their names, data types, dimensions, descriptions that we use in the data model. And so a core concept in the specification language is the idea of NUR data types. If you're familiar with object-oriented programming, you can think of a NUR data type as a class. So NUR data types define high-level data types such as a time series that we can then reuse through inheritance and composition. And so this is a concept that you will see coming up also in the APIs, where distinct types of data are represented by specific classes in the APIs as well. And then, as I mentioned, NLB also supports the concept of extensions of the data standard schema using the same specification language and rules. And the data standard schema then uses the NLB specification language to define a large collection of these reusable NUR data types for storing a large set of neurophysiology data types and to define their organization as part of the NLB file hierarchy. And so one core concept that you will very quickly come across in NLB is the idea of a time series. So a time series defines a very base type for any type of time series data and recording, and it then breaks out into more specific types such as electrical series for an extracellular recording, image series for an optical physiology recording, or patch clamp series for intracellular electrophysiology recording. And so just to give you a general idea of some of the basic concepts we use in the NLB standard to organize neurophysiology data, we'll have a quick look now at how extracellular neurophysiology data is organized in NLB. And so first off, you have general metadata about, in this case, the electrodes that are used in recordings, which is stored in a central data table as part of the general metadata of the file. And so here we also then store metadata about the devices, the subjects, and other parts. And so the first concept that you see here is that metadata is commonly organized for us in tables that are then also interlinked with other types. And then recordings of extracellular electrical signals are then stored in electrical series as part of the acquisition group. And so the electrical series then also explicitly references again the relevant metadata, in this case to define which electrodes specifically were used in the recording of a particular electrical series, as you may have more than one electrical series recorded from different sets of electrodes. And then NLB also supports storage of primary pre-processing results here in the form of detections of spikes and other events and features in the data, which are stored as part of the processing group in NWB. And then finally, in the context of extracellular recordings, NLB also supports storage of the results from spike sorting via the units table. And again, NLB here explicitly links the data back to the relevant metadata descriptions in the electrodes table and other places. And so NWB similarly supports storage of data from optophysiology pipelines as well. The basic organization is similar, where you have general imaging metadata, and then you have acquisitions of raw and motion-corrected time series recordings, which are stored in the acquisition folder. Then you have primary processing via image segmentation or detections of regions of interest, which are then stored in the processing folder, as well as derived neural activity, similar to what you have here for the spike traces. But in the case of optical physiology recording, those are then fluorescence traces or DFOF traces that you will store in NWB. And so NWB provides neurodata types for most neurophysiology recordings from extracellular and intracellular electrophysiology, optical physiology, behavior, stimuli, and experimental metadata. And so if you want to know more about these specific types, then you can look those up in the NWB specification or in the APIs directly. Or if you have any questions, you can always reach out to us on Slack and the help desk as well. And as I mentioned earlier, in addition to the types that are supported in the NWB core schema, we also support the concept of extensions, which use the NWB specification language to define new types of data and metadata. And so here's just a very quick example of an extension for storing the cortical surface of the brain. As you can see, PyNWB provides us with a dedicated API to create an extension schema. It looks very similar to how you would create a file, only instead of explicitly creating a group or a data set, you're now specifying how that group and data set need to look like. and then you simply export that information, and it creates a YAML file that with that schema. And once we have that schema, we can immediately load that namespace with PyNWB, and using the get class method, we can then automatically generate container front-end classes to populate the data in memory and write it to disk. And then similarly on read, we can again load our namespace and then read that data back in, simply based on the schema itself. You can always also generate your own front-end container classes, but for initial exploration, this should work out of the box. And then similarly, MATLAB supports the same kind of process, only here you will call the generate extension function first to generate physical MATLAB code for your extension. Then you can read and write that data in the same way. And again, PyNWB and MATLAB are both interoperable. So if you generate a file in MATLAB, it will be readable in Python and vice versa. And then to facilitate the community to adopt and curate NWB and integrate new use cases, NWB provides easy to use tools and processes for not just creating extensions, but also for sharing and evaluating, reviewing extensions. In addition to the extension API that we looked at briefly here, there's also the extension catalog, which is an online catalog for sharing and reviewing the extensions. There you will also find the NDX template, which is a cookie cutter based template to allow you to easily set up and create new repositories for creating extensions to make sure your extension follows best practices. And then you can submit those extensions to the catalog through the stage extension repos. And then in addition, there are a lot of core tools such as the HTML documentation utilities to allow you to easily document extensions, as well as a broad range of policies with rules and guidelines around how to handle extensions. And though using these tools, we're working with the community in community working groups to further develop the NWB data standard. And so there's a broad of ongoing community working groups, such as on fair representation of genotype, spatial coordinates, behavioral tasks, and so on. And you can all contribute to these working groups, and you can all contribute to extensions, and let us know if there are certain things that you need or that you believe the community will need, and just reach out to us again on our help desk or Slack if you have any questions on that. And so then using the concept of narrow data extensions supports curation of NWB standard with the community. We can fully test extension proposals before they become integrated with the NWB data standard, so that we can feel confident that any new data types that we make accessible to the community have been fully vetted and tested. And so then the next key component in our ecosystem is the data storage component. The data storage really just looks at how can we map the primitives from the specification language, a group, a data set, an attribute, to actual storage. And so NWB currently uses HDF5 as its main file storage backend. And we had chosen HDF5 because it supports large-scale storage of complex data collections in a single file. It is optimized for performance, supports parallel I/O, as well as a broad range of advanced I/O filters. It is supported across platforms and programming language for MATLAB, Python, C++, Fortran, R, and many others. And it targets long-term support, which is really critical for a data standard such as NWB. We're also working on exploring, as part of PyNWB, other storage options such as ZAR. But the primary backend that you will encounter right now is HDF5. And through HDF5, NWB supports a large range of different advanced I/O features that will really help you interact with your large-scale data. We support lazy data load in both MATLAB and NWB, which allows you to open even very large data files without having to load all the data into memory. All APIs also support chunking, which allows you to optimize the layout of your data on disk. We support I/O filters such as compression to reduce your data and storage cost, as well as NWB supports self-contained data storage, where you place all the data in a single file, as well as modular storage, where you can distribute data across multiple NWB files. And then finally, we also support iterative data write in all APIs, which allows us to stream data into the file for example, during conversion, if you have really large files on disk that won't fit in your memory while you convert, you can do this through iterative write processes. And then finally, we have our data APIs, which are PyNW for Python and MatNW for MATLAB. As I mentioned, the APIs are interoperable. That is, NWB files that you write in MatNW can be written in PyNW and vice versa. And both APIs support efficient read, write, and query and analysis of NWB files, including support for streaming, chunking, and compression, as I mentioned before. And then to learn more about PyNW and MatNW, we have our tutorials online. As part of the APIs, there are domain-specific tutorials that you can go to if you're interested in a particular area, such as electrophysiology, as well as general tutorials that will allow you to learn more about NWB itself. We also have a training course up on the INCF training space, as well as all the recordings from our previous user training sessions are available on YouTube. So there are these resources available if you want to learn more about NWB. And if you run into any specific questions, is that you can go to our help desk or to the Slack channel. If you go to our help desk, don't be surprised that there's not a lot of messages there yet. We just created our help desk on GitHub. Previously, we always did our discussions on Slack, but we encourage you to use the help desk on GitHub that we have now to make sure the discussions are open and accessible to the community. And so a key goal of our efforts in NWB then is to help really the community overcome the energy barriers in data standardization and to advance fair neurophysiology data through dissemination of NWB. And so working with the OpenScope project is one key piece of this as well, to make sure that all the data that is generated by the observatory is easily accessible to the community. And so one thing to keep in mind when we look at the energy barrier is that it's not just a technical challenge, but it's in many ways driven by cultural challenges. And so the cost of adopting standards is not just about converting data into a particular format, but it's about how do we in a lab or project and across the community want to work with each other. And so the DANDI archive also provides a very good way here to allow us to collaborate on data together. And the OpenScope project is really a leader here for us in the community to show how this can be done across a project like this. And so on the one hand, there's different approaches that we can then use to help the community overcome this energy barrier. On the one hand, there's the STIX approach that provides a framework, support and motivation for users to climb the barrier to adopt NWB, to adopt standards in general. And so we do this through engaging, for example, with NIH, International Brain Data Archive, such as DANDI and other stakeholders, to help really create, on the one hand, the funding incentives, but also the data governance principles that help us create an environment that is conducive to standardization. But the key approaches that we take then at NWB are really about guidance and support to help lower the barrier to adopt the standard, as well as provide immediate benefits and impact to you via reduction in cost, time, and effort, improved quality and reliability and accuracy of results, and access to new scientific capabilities. And so as such, a main aim of our efforts really enable the neuroscience researchers to efficiently utilize NWB and the associated software with the goal to lower the barriers of adopting NWB as a standard and promote broad dissemination of NWB by focusing on the needs of the neuroscience research labs. By, on the one hand, creating training resources and providing in-person training opportunities, by providing support for labs and projects to integrate their data and processes with NWB. So we're also providing consulting to the community. And so if you're interested in talking with us and having a consultation, then please reach out to us on Slack or the help desk and let us know, or contact me or Ben Mendictor or any of our team members by email, and we can also set up consultations that way. And then the last part here is that we also create then the community-driven processes for integration of new use cases with NWB through our catalog. And then over the last years, we have seen significant growth and interest in adoption of NWB. And so, for example, in 2020, we had more than 600 participants from around the world at NWB training events. In addition to training events, NWB also provides a broad range of online resources, such as all our online documentation, video tutorials, and then our community channels through Slack and our help desk online. And you can find all these resources online on nwb.org. And I also included the slide at the end to give you an overview of everything that's available. In addition to neuroscience users, a key target for our outreach and dissemination activities are developers to promote and support integration of NWB with neuroscience tools to make sure that all the tools that you need are available to you and that they support NWB. And so our goal is really to create and maintain a vibrant software ecosystem around NWB and to create incentives for you as the researchers to adopt NWB by providing the right tools to you. Key components of our efforts to enable the integration of NWB with neurophysiology data analysis and management tools are to provide support for software developers to integrate their data acquisition, processing, visualization, and analysis tools with NWB. Again, we provide consultations to developers of software tools as well. So reach out to us if you have a need for one-on-one consultations and in that regard as well. And then maintaining our core software ecosystem to make sure our software is stable, reliable, and really ready for production use is a key piece of all the work that we do. And as you can see in the plot on the slide, we continue to exhibit significant growth also in our software ecosystem. And so as such, developing and maintaining our ecosystem is a key piece of what we do. And so this includes that we're enhancing the accessibility of NWB through best open source software practices, documentation, and engagement. We ensure reliability of our software through careful testing, continuous integration, and code reviews, and many other technologies. We ensure stability of our software through careful design, for example, to insulate the different aspects of the ecosystem that we talked about, and really to support the community through broad range of our community channels and documentation, as well as to enhance our tools to make sure they work properly with all the tools that you need. And so in conclusion, going back to our original motivation, how does NWB solve problems in data sharing? Well, we said sharing neurophysiology data is tedious. Well, with data in NWB, the data can be understood by anyone that is familiar with NWB. They don't have to be familiar with any other of the standards, and you have all your data accessible in one container and file. And if you share them through DANDI, finding your data becomes even easier, and it's also much easier to use because you have tools such as the DANDI Hub available to you as well. We said data have limited reuse outside the purview of the original experiment. Again, data in NWB is designed to have sufficient metadata to be reused. And it's really exciting when we see folks picking up data from others and applying their methods and integrating their own data with NWB. And through the OpenScope project, I think we will see many of these sort of experiments happening where it's not just you using the data that you generate, but other people will also want to use the data that comes out of the OpenScope project. And then performing analysis and visualization requires converting data from one format to another. Again, here NWB seeks to provide you with a software ecosystem that allows you to keep your data in the NWB data standard from the time you acquire the data to the time you publish the data, and perform everything from your processing and analysis all in one coherent standard. And so finally, I want to thank the entire NWB team, as well as all our NWB users and developers. NWB is truly an endeavor in multidisciplinary team science, and NWB would not be possible without the contribution and support of the community. And so as we move to our questions section, we have Ryan Lee from LBL as a core developer on the phone as well. And then Ben Dichter from Catalyst Neuro, who is also the PI on our current NIH grant that supports development, and NWB is available as well. And so Catalyst Neuro provides support to neuroscience labs, but if you have questions about a Catalyst Nero, Ben can introduce that in a minute as well. And so here, just for your reference, a list of all the various online resources. Again, you can find all of this information on nwb.org. So visit us on our website. And with that, I'm open for questions. Ben, do you want to say a few words about what you do as well?
Sure, I'm happy to. So I'm the founder of Catalyst Neuro, which is a software consulting company that specializes in helping labs adopt NWB and publish data on DANDI. As Oliver mentioned, we recently, he and I are the lead PIs on this grant. It funds us to basically offer consultations and support to individual labs, and we would be happy to work with any of you listening who are interested in adopting NWB and don't really know where to start. We're happy to talk with you and discuss how to build conversion scripts and publish the data.
Thank you guys both very much for that overview. It's a great introduction. It's like we said for Satra as well. This is a great introduction as what the resources we use for OpenScope, but also beyond that in OpenScope, just for the broader neuroscience community. We encourage everyone to leverage these resources for their own projects as well. We have time for Q&A for this group. So if you have questions, please enter them into the box. And just a reminder that this is recorded, so all those resources that Oliver listed of how to reach out and get in contact, you can have as a reference in the YouTube if you need to go back and find those again. And please feel free to reach out to us too if you need anything forwarded to their team as well. Just like we said for Satra, if you guys have specific questions for NWB and how we'll leverage that for the OpenScope, please enter a note into your individual consultations so we can see if we can forward and answer those questions in advance from like Oliver and Ben. I had a few questions to follow up to what we asked for Satra as well. It's just opportunities to learn more. And you listed a lot of resources in different trainings, but are there similar either virtual or in-person events you have planned for the upcoming year of just how to engage more with your team?
Absolutely. So we typically aim to have at least 2 in-person training events that we host ourselves. So this year we had our developer event in April, and we just hosted our user days at INCF a couple of weeks ago. We're looking into having another event coming up soon, focused more around documentation. Again, we will have a user-focused event and a developer-focused event coming up again in probably spring of next year and then summer of next year. We haven't announced those yet. We're still planning the exact dates. And then usually you will also find us at various conferences, whether it is at SFN or INCF or the Neuromatch Academy, where we'll provide different forms of tutorials. When we have our user days, it's typically a 3-day event where we actually really work with the people, not just providing talks and training, but where you bring your own data and you really have your own hacking project as well. Where when you're at a conference, it's usually more about the direct training, and then you do your projects at your own site. So we provide all of these events. I don't have specific dates right now for the upcoming ones yet, but just keep an eye out. We always announce them on our nwb.org/events website, and then if you sign up for our newsletter, you will also see announcement of those events there. And don't be afraid to sign up for the newsletter, where we're not sending out emails on a monthly basis. It's usually every 3 to 6 months that we send out an update newsletter to the community, depending on what new dates we have available. So it's not a regular spam. It's really just we send them out when there's something really interesting to announce.
I was also curious to hear more of just the motivations, the carrot and stick, as you mentioned, both in either things you see up and coming as far as funding incentives from like the NIH to push to use this more, or examples of collaborations that happened because of this that maybe wouldn't have before. So I'm curious if you have anything that was good motivators for others or just examples of success stories.
Sure. I think I'll let Ben take this question.
Yeah, sure. So there was a notice maybe a year and a half ago from the Brain Initiative that I think it was after March of 2020, all of the proposals for the Brain Initiative are going to be required to have as part of their project to convert data to the appropriate standards and publish it on the appropriate brain approved archives. So for neurophysiology, that would be NWB and DANDI. So projects that started funding in January about, they kind of have this new crop of projects that are being held to this new standard. And so we're really building the infrastructure and starting to communicate with those labs that have this new requirement. And more labs are going to have that requirement as the funding cycle kind of hits those new projects that are funded. So that's specific to the Brain Initiative. It's not NIH wide yet, but my understanding is that they're looking at kind of testing this requirement out on the Brain Initiative and working out the kinks, and then on a kind of a few years timescale, perhaps expanding it to NIH wide. So there really are quite broad funding incentives being put in place that are kind of pushing people in this direction. And then, of course, that's the one side. The other side is all of the tools being developed. I mean, the pace at which DANDI has been able to develop their ecosystem, it's really, as you saw in the talk, it's really much more than an archive. It's a whole platform for collaborative research and computation on the data as well with the DANDI hub. So the idea that we now have free compute that people can use to analyze data that you're streaming into that compute instance, and then we have a whole slew of analysis and visualization tools like Suite2P and Cayman and Cell Explorer and Spike Interface. A lot of really state-of-the-art processing tools are being integrated with NWB, and we're working with more, like we're working with Deep Lab Cut, which is very popular now. So not only can you have this compute for free, you can also access state-of-the-art resources that we will kind of work with them to make sure that they work effectively in that cloud setting. So, I mean, I think from the side of it being useful for you right away, I think we're really quickly getting to the point where it's worth people's effort to convert to NWB because you're actually able to access all of these tools. And then on the other side, it's worth your effort because you either are being held to this standard or you likely will soon be held to this standard. So you might as well get working on it now.
Yeah. Another thing to mention is it's also, over the years, we had very great support by also nonprofits such as the Kavli Foundation, as well as the Simons Foundation, who are continuing to fund efforts to help people convert data into NWB. So for example, if you go to the Kavli, they're putting out seed grants to help labs adopt NWB. And then more broadly, we're also reaching out, to other brain initiatives internationally to work with them on getting NWB adopted. I mean, open source brain is just one example of an archive that sits in Europe that is supporting NWB, and so it's really growing, not just as a scope within the NIH and the US, but we're hoping to really grow it as a really international standard for neurophysiology.
So already aspects of there's the standard somewhere with NIH and very likely to grow in the future. So it sounds like all the resources you have to learn about it are good to take advantage of right now. You also mentioned, I'm just looking at ways for people to get in touch and get involved, some of the working groups going on. Can you say more about that or how people can get involved with that?
So for the working groups, the easiest way to get involved with is either, for the ones that we're hosting directly, just reach out to me directly. Some of the working groups are also hosted by INCF. So there's usually clearly a lead of that working group indicated that you can reach out to, but you can always reach out to us directly to do this. Similarly, these working groups are typically driven really by very specific use cases in the community, where users come to us with a large problem that they have to say, this is a general problem community, we need to address this, and we need to address it in a way that's not specific to a particular lab. And so if you find such a problem, please approach us about it, and then we can see, who would be the right members to start talking in the community. It is really, our role is to be the facilitator at that point, to make sure that we can set up these working groups, that we have the right people in the room to bring the community together. And so any issues that you have, or even if you have created an extension that you say, this would be ready for bigger use, then submit it to us on our catalog and let us know about it. And we can have a community review of these kind of things. But the easiest way is just reach out to us on either our help desk or Slack, and then we can see that we can get this going. The goal is to have as many people involved as possible. So we're always open to have you come to our working groups.
Great. So don't be shy about reaching out to you guys. And again, the contact information was on one of the slides if people need to look back at that as well. But just the website is nwb.org, right?
.org, and you will find the links to our Slack channel and the help desk right on the front page.
Perfect. And one other question, we talk a lot about NWB with the DANDI archive, but what about it interacting with those other archives that are out there as well? What kind of interaction, or how do you see those two?
So as I said, so one other archive that's already supporting NWB would be the open source brain. DABI also supports NWB. We're open to work with any archive at the end of the day. People are also using more generic platforms such as Figshare to share the data, although I would always encourage to use a platform such as DANDI because of all the additional features that it provides to make the data really findable and accessible. Because just because data is available in a standard doesn't mean you can easily find it. Right? Just because a link on Figshare doesn't make the data easily findable. It's all these other features that an archive like DANDI can provide that allow you to then go in Google and actually search for data and really understand what the data is about before you actually download it. But right now, from an NIH perspective, DANDI is the primary archive for all of neurophysiology for us.
Great. Anything else either one of you wanted to add that you didn't get time to brag about or touch on before upcoming updates? And then any questions or comments from the other panelists related to NWB or DANDI they wanted to add or ask?
Yeah, but I have a question for maybe Satra and broadly. So one of the dream that we could sort of bring to the table is by having this 14 projects all through NWB and DANDI, and then potentially all using the standards. At some point, like a team could join and say, "Hey, 13 other projects. Do you have the code for this figure? I want to retest exactly that on my own data set." And so I was wondering if you thought about how to have a standard of repository code for analyzing all of this. So I'm sure, Ben and Satra, you have thoughts on that.
I sure do, but Ben, do you want to take this on because you've actually done it for a given DANDI set?
Yeah. So we are currently developing the infrastructure to do that, and a lot of the pieces are already in place. So I worked with Bing Brunton's lab, and they had Jupyter notebooks that could reproduce panels of figures for recent publication. And the data from that publication, we published it in NWB on DANDI, and then we used the streaming feature that Satra mentioned on the DANDI hub, and we were able to share notebooks that reproduced the figures from the paper in a way that anybody who signs into DANDI hub now has access to those notebooks, and it's associated with that DANDI set. So this is our we only have that for one paper right now. It's kind of a proof of concept, but yeah, we would want to expand this to many more papers. We see the ability to reproduce the figures of a paper to be a really really incredibly valuable feature of the archive and of the compute infrastructure around the archive.
More generally, I would say, I think we want the ecosystem of tools to increase, right? And the more they can be put into libraries that could be reused, that's actually, I mean, there's a little bit of a software engineering perspective to this as well, beyond kind of the notebook perspective. So if we can get algorithms consolidated in some standard libraries, it just helps improve a lot of things and maintainability of those resources. So while the notebooks are a point, it perhaps is an accessible point to many individuals, I think there is a broader push that we should, as a community, make towards kind of encouraging library-based reuse.
And
Yarek.
I sneak in as well. Hi, I'm Yarek. Getting back to which other archives support NWB and integration, and sneaky us or others, right? That's the beauty of sneaking in into standards, that as long as standard supports another standard, like bits supports NWB, then OpenNeuro, which supports bits, is in principle already supporting NWB deposition, right? If there is proper combination of domain and modality recording appropriate for OpenNeuro archive. And also with bits, you gain standardization of some code location. Let's say there is code directory reserved that for that study, that's where you would put some pre-processing script or you would put an anonymization script. So then it becomes integrated within the data set itself, which is, of course, doesn't replace creating libraries, which is much more better for use, but it gives you exact point of, you know, this data set, this is the code which comes with it, and you know where to look for it.
One thing also, you know, looking at the OpenScope project, the nice thing is, you know, it allows us to now think beyond just, you know, here are individual Lego pieces, individual tools that become accessible, but it allows us to think about what is the actual pipeline from beginning, from acquisition to publication that you want to use, where you say, this is how you will process your data, this is how you analyze your data, so that you actually have the full end-to-end pipeline available. And I think that is really something, if in the next few years, if we can have some standardized pipelines for optical physiology, electrophysiology, and so on, that will really help the community as well to really drive forward standardization.
Great. I don't want to put a pause in this discussion. I think we should continue it with more time, but we do now have time for just any questions related to the OpenScope program, the RFP, DANDI, NWB. If anyone in the audience wants to, again, use that Q&A box to ask additional questions on any of the topics we talked about today. But we do also like using these webinars as kind of community building discussion. So if panelists do have additional comments, I encourage you to keep the discussion going. But any questions are welcome now. And again, if you have more questions afterwards for those individual consultations, let us know or reach out to us on openscope@alleninstitute.org. And I do, just to start that off, looking at how NWB and DANDI, again, are going to integrate with these specific OpenScope projects we are awarding. I think Jerome or anyone to clarify how much the awardee is expected to know what to do for NWB and DANDI versus how much we're setting up for them. Maybe, Jerome, you want to start that off?
Yeah, so we're committed to convert all experiment to NWB files from the get-go. So the goal is to have them available nearly immediately after data collection. So you'll be seeing those files embargoed as real time as we can on DANDI. And so then you can use the PyNWB resources to access those files. I'm hoping some of the widget might work out of the box. I'm sure Ben can help you guys support it. We also have some staff with the resources to help you do some analysis. So sort of to help you access those files. We still have to hire those people, but they will come on board to help with the process.
And Jerome, maybe I can ask / kind of state a couple of things, which is given the kinds of data you're generating, it almost seems like there are some standard workflows for pre-processing that could be run on the raw data almost immediately, right? And so again, that would be of advantage to many labs who are doing this, because they won't have to, they can always dig at the raw data, but they might get better derivatives out of it. So that's something I think that could be worked towards as well as for any of these projects. And then the question I had was, I know there's an initial process of quality control that I think you plan to interact with the projects on. Is there a useful setting in that process to upload the data to DANDI, even at that initial, before you launch full-scale collection, for people to kind of use DANDI to look at the data and play with it right there?
So you mean, I think if we can add sort of quality control metrics to the NWB files, what you meant?
So just, I mean, I can't imagine all possible sources of quality control, but let's say you run the experiment on a couple of animals, right? Just to demonstrate feasibility or other things for a given project. Is there a step in, I thought I saw somewhere in the pipeline, I think there was a step of kind of doing an initial collection before launching the full collection. So it almost seems like that there is a potential to integrate with project teams at that point of that initial collection, and that could be done, at least for the digital part of it, on an archive like DANDI with NWB.
Yeah, so I think our current plan is data set that are collected during the pilot to validate should be converted to NWB files and uploaded. So that will be our valid. So we will run the QC metrics internally. I'm hoping we can add some of them to the files itself, but then the ones that passes will go up. And so this way, the external team can sort of have a first view into the data. My personal goal is to all interactions will only go through NWB files. We will not sort of request raw data access. This is something that we learned from the past few years, which we would like to avoid that dependency on the way we store things internally, because this can change. I do want to make a couple of points that were missed in the talk this morning. One of them is about authorship. It's important the teams that make proposals, we guaranteed first and last authorship slots. So obviously there's a project is a contribution from a lot of people, but the applicant team is as that ownership guaranteed. There was also a point about, I think we also a community project. OpenScope is a community project, and so all of this experimental capabilities are subject to change if there is a big community interest. So for instance, the opt-in, general opt-in questions, we have done some of these internally, and so if there's a big interest, we could look into that in future years. But obviously that's a lot of work, but we'll have to look into that. There was a question on anesthesia. I encourage a person that is interested in doing that to reach out. We can follow up on that. That's the things I had noted.
Yeah, thank you, Jerome.
There was also a question in chat by Bhavania.
I was just going to tee that one up unless there's more specifics before that, but this seems like a very good discussion question. So I encourage you all to give your input on this. But there's a question of, in this whole picture, how does standardization across all these realms limits creativity and creates limitations in the research process itself? So this is quite a good discussion question if anybody wants to jump in on this one.
So my general view on this is the goal is quite the opposite. The goal of standardization is to free up energy to be creative, right? Because you don't have to worry about all the pieces that are already standardized, you don't have to think about. So you should have the ability to only think about the pieces that are specific to your experiment, that are specific to what you need to do. And as I mentioned before, for NWB, we support this sort of creativity by allowing you to take what we have on the standard and building upon it by creating extensions. If there's specific data that we don't support yet, then you can create an extension very quickly to add that information into the file. but you don't have to worry about all the things that we have already worked for now a decade trying to figure out how to store properly. So my answer is the opposite. It should free up energy for creativity once you have adopted a standard.
I mean, I can comment. So I think experimental exploration is also a big aspect of normal research, and I think one should see the observatory model as complementary to the existing way research is done. And so in some cases, you might want to have total control on the experiment and explore parameters that are impossible to do at larger scale. And so you just fine tune the experiment and figure out things that are working. And then once you have this figured out, you partner with the observatory to do it at sort of high standardization and perhaps a larger scale. And so I think these are complementary one another. The experiment that are impactful in an observatory context, I think we should have this dialogue with the community and integrate them into the platform. But some of these might not just be one experiment off. And so in this case, maybe they're better in a traditional lab. These are complementary models, and so they shouldn't sort of see as against one another. So I see it.
And Jerome, correct me if you want to refine this, but this is kind of one of the questions in OpenScope too, is how can we leverage the standardization at the Allen Institute and reproducibility and reliability that comes with that, but start adding in more hypothesis-driven creativity while maintaining those existing standards at the Allen Institute. So that's kind of the key question here, and looking for you as proposers to find creativity within the constraints we listed in the RFP and to suggest something that works within the existing pipeline. And as we see successes there in the future solicitation cycles, how we can start ramping up additional creativity or changes from that base pipeline model. So I think that's a great question for this opportunity as a whole.
I mean, and what you get at the end is not a limitation. You get 6 probe neuropixel recording and height scale with like everything in the standard that interface with previous projects. So you actually get much more than just that project. And so these limitations are it's just entering into the ecosystem, and then you open up a lot of other things.
Anyone else want to add to that bigger picture question? Or have specifics that they didn't get to mention in the talks that have come up since then?
I could add a little bit from the DANDI side. I think Oliver used this term, the descriptor, the language of neuroscience in the context of NWB. I think with all the tools and evolution of hardware, science is an evolving process, and I think this speaks towards the standardization. We want to encapsulate things that improve our efficiency, not having to think about it because we've done certain things, and then kind of look at things where we need to evolve. So even right now in DANDI, for some of the microscopy data, we're still organizing and working with the community to try and figure out the best ways to encapsulate it, to compress it, to reuse it. This is not an experimental question. This is more of a technological kind of question. And that's going to continue to evolve as we work through those things. But in terms of standards themselves, and this is a balance that anyone who designs standards kind of deals with, is the science is moving. And it's unlike, I think, a typical standard in the industry where you put it out there and then everybody uses that particular standard for a time moving forward. Here, it's really more about where we have adopted efficiency by using standards and then increasing the capacity of that standard to extend out to different scientific questions. I don't know if that question was more on the experimental design side of it, given the restrictions of the rig and other things, as opposed to, and I'll give an example from a slightly different domain, neuroimaging. So in many of the cognitive tasks that people do, people design a very specific cognitive task, and then they would do neuroimaging while that person performs this task. Then we kind of moved on to doing resting state analysis, which is people just lying in the scanner, not doing a specific task. And then there are people who are watching movies or playing games in the scanner, something a lot less constrained than an actual cognitive task, but a lot more flexible and more realistic in certain ways. So if I could extend it out and we could all have helmets that were MR scanners, then imagine just collecting data as you live your life. And that's happening with certain devices, right? Fitbits and other things. So there is this space of science, which is from complete control on one side and being able to explore in that controlled space to complete exploration and then picking out segments of that exploration that might be relevant for scientific questions. And I think that's the kind of thing that we're always dealing with across various broad domains of experimental science. I can't speak for OpenScope, but I do think as scientists, we have to be able to play around with all of these possibilities. And one should not see standardization as a limiting factor in these possibilities. It's just kind of supporting the exploration of some of these things.
Great. That is well said. Great. Well, I do not see any more specific questions coming in from the audience, but last call if anyone has anything to enter in the Q&A. But a reminder that this is recorded and will be on YouTube if anyone wants to see the talk after this or share it with a colleague or collaborator. And then please, again, reach out to us at openscope@nlsu.org if you have additional questions or have questions on the RFP or want connections with the Neurodata NWB team or the DANDI team as well. Unless anyone else, Jerome was raising your hand.
Yeah, before we leave, I do want to thank Carly for organizing this and leading the effort. Was a really great lead on, as well as the communication team behind all of this. Jenny, Megan, Caitlin, and others that really have put this together. And also Satra, Ben, Ryan, and Oliver for being there. It was really great to have you.
I'll share that. Thank you guys for your talks, and thank you for the comms team for being behind the curtain there. All right, with that, I think we will sign off, and thank you guys all again.
Thank you.